an esteemed gathering of researchers, scientists, and microbiome enthusiasts here today. We welcome our dignitaries for today, Professor Raghunatha Rengasamy, Dean GE, and Marty Manaria Gurunath, Chair Professor, and Professor at Department of Chemical Engineering at IITM, and Professor Ashwin Mahalingam, Dean of Alumni and Corporate Relations, and Professor in the Department of Civil Engineering at IITM, to the dais, along with Professor Karthik Raman and Professor Karthik Anantharaman, organizers of this symposium, and also Professor Kasturi Venkateshwaran, who's adjunct professor at Department of Space Studies, University of North Dakota, and Satish Dhawan, professor at IISc Bangalore, and also an adjunct faculty at IITM. We welcome you to the dais.
Arrange the chairs. Let me just get started. Good morning, everyone. Really happy to be here at the 5th IBSE International Symposium. IBSE started off in 2015 as a small initiative, and now it's grown to a fairly good center, which is playing a key role in several national and international projects like Genome India, Incentive, Metasub, the Garbhini project on maternal and child health, and so on. So we were established with the goal of trying to leveraging IIT strength in quantitative sciences and bring it to apply on biological problems. So we're very happy that this has taken off the way it has. This is the 5th edition of our symposium. The first one was in 2018. Then in 2020, we had an EMBO symposium just before the pandemic struck. So all our speakers have very fond memories of our IBSE symposium, which was here and in Mahabalipuram. We are going somewhere closer to that tomorrow for our excursion. And then we had something online during COVID. And then the 4th symposium was organized by Venkat and myself. It was again on the microbiome theme. This was during November 2022. So this is our 5th edition, and we are very happy to have a wonderful gathering of speakers here, including a lot of early career researchers, which is something that we've been pushing hard for, and you'll see them being session chairs and so on, getting a lot of opportunity to talk to all the other folks who are here. I would like to first acknowledge some of our funders. So first and foremost, I would like to acknowledge Prakash Arunachalam, who is an IIT alumnus of IIT Kanpur, who had a very generous endowment for the center, which has helped us fund a lot of activities, not just the symposium, but several other activities. And I'm also very grateful to the Office of Global Engagement for funding this conference. It really helps us pay all the travel bills and so on. And I'm also very grateful to the past deans, Professor Nagarajan, Professor Mahesh Panchagnula, who've been very helpful in getting the initiative going, and also to our current deans who are here, who've been very instrumental in getting many IBSE activities going on. So at IIT Madras, we have a really supportive administration, and you'll hear a little bit more about some of the things that we do to enhance the research atmosphere and internationalization at IIT Madras. And I'm also, very grateful to all the participants who are here and our sponsors for the symposium. I hope we'll be able to attract more sponsors next time. I think people are still catching on to microbiome, but very happy that Hindustan Unilever has funded us for the symposium, and Taylor & Francis has also agreed to kindly sponsor the poster prizes that will be announced on the last day. And with that, I'm really happy to welcome you all, and I would now like to invite my co-organizer, Karthik Anantharaman, to share a few words to introduce the symposium. And thanks, Karthik.
Thank you so much, Karthik. I'm just going to keep this very brief, but to introduce myself, I am an associate professor at the University of Wisconsin-Madison. My name is very similar to Karthik. I'm Karthik Anantharaman. There's a big story behind how we interacted with each other and how we have gotten here. I have a little bit of time, so I'll just say, but our names are similar. People do confuse us for each other. And this is not just regular people. It's journals, editors, conferences, everywhere. So we get each other's acceptances. So we decided if that's the case, people are confusing us for each other, why not collaborate and bring more confusion into the mix? So that's why we decided upon this plan. But just to say, we are very excited to have all of you here. The idea of the symposium is to truly bring together an international group of scientists collaborators, early career researchers, students, postdocs on a diverse field of microbiomes. One thing I want to mention is that this symposium is going to be regularly held. Karthik and IBSE and IIT Madras have this vision to have this every year. So if you are thinking about a symposium that you'd like to attend in the future years, if you're here in India, you want a symposium focused on microbiomes into the future, this is something you should consider. I want you to appreciate that this is truly international in nature. You should appreciate the diversity of speakers that we have and the diversity of topics that are going to be covered over the course of the symposium. We will have talks on viruses, on environmental microbiomes, different kinds of environmental microbiomes, plant microbiomes, insect microbiomes. There are talks on evolution, there are talks on archaea. So there's talks on extreme environments, including space. I truly want to appreciate, I hope you all can appreciate the diversity of topics that will be covered over the course of this symposium. The setting here is pretty small and intimate. This is by design. There's about less than 150 participants. The idea here is that we want to keep it small, intimate, so that students, postdocs, early career researchers, you can talk to scientists and gain both ideas as well as the experience of interacting with them. And lastly, I want to say that there's not just scientists here. We have folks from industry who are here. We also have Sahana Ghosh from Nature India. Sahana, if you're here, you could just raise your hand. Please reach out to Sahana if you want to talk about things beyond science, communication specifically. So the idea here is, it shouldn't just be about science. At the end of the day, communicating is important. Talking to industry is as important as talking to scientists. That's the idea that we are trying to bring about. And I'll just say that tomorrow, as Karthik alluded to, there is an excursion to a heritage museum called Dakshina Chitra that happens in the afternoon. So the idea is that we are all here to discuss about science, but we can also take it outside in an informal setting. So with that, I would like to welcome you all again. Thank you so much for coming, and we are all looking forward to an exciting 3 days ahead.
We call upon Professor Raghunathan to also say a few words.
Thank you very much for being here. It looks like an occupational hazard of a deanship that I address all conferences in IIT Madras. Also the fact that our office supports these conferences, and in fact, Karthik helps with this, and I actually don't do much. Most of the work is done by Karthik. But in any case, very quickly, a few words about IIT Madras. For people who are here for the first time, this campus is about 625 acres of wooded land. You can see deer, monkey, and also one peahen. Suddenly it made its way somehow into the campus. And we are about 10,000 students, 3,000 undergraduate students, 4,000 at the master's level, and about 3,000 at the PhD level. We have about 600 faculty, so that makes it roughly 5 PhD students per faculty. Last year, just to give some numbers, we published some 3,500 papers, raised about 1,000 crores in funding. And so just gives you an idea of what happens here. And of course, IIT Madras in recent years has been really pushing on innovation and taking things to market. We had news yesterday that one of our startups has got an order for some $1 billion order for about 750 electric vertical takeoff and landing helicopters for medical purposes and so on. So that's broadly IIT Madras. In terms of our Office of Global Engagement, we look at working with universities, companies abroad. We are involved in setting up campuses abroad. Right now, one singular campus in Zanzibar. And we look at organizing international conferences and so on. So that's broadly the vision of our office. The last thing that I want to say is I feel a little sense of sadness when I see words in conferences where I have done some work and I'm here speaking as a dean and not anything technical. But that's again an occupational hazard, I think. Nonetheless, I'm very happy that Karthik have put together a fantastic program here. Happy to welcome you to IIT Madras, and hope all of you have a great 3 days of productive scientific discussions. Thank you.
We call upon Professor Ashwin to also address the gathering.
Hi, good morning, everyone, and welcome to IIT Madras. I am here because I'm the Dean of Alumni and Corporate Relations. So as Raghu said, I also am part of this group of people whose occupational hazard includes coming and talking about these, talking to people who attend these conferences. Unlike Raghu, who I think was hinting that he may have done some work in this area, I'm a civil engineer. So we build massive structures that can be seen from long distances. Microbiomes are probably the exact polar opposite of what we do, but it's sort of great to see so many people from all across the world here. So congratulations to Karthik and Karthik too. I think that's now starting to become a bit of a theme now, putting this together. Just wanted to say, I think that, despite the fact that my background is very different, before I took over this role as Dean, I was heading something called the School of Sustainability that IIT Madras had started. And the School of Sustainability was, in our view, is a little bit different from several other schools that have been set up elsewhere in the world, because ours was not set up as a department. Ours was set up more as a platform. So the idea is we have 18 different departments at IIT Madras. Everyone has something to do with sustainability. Can we bring people together from across departments to really work on some of these large challenges? And when I was, just quickly Googling a little bit of what microbiomes were and what they do, etc., one of the things I came across was a whole series. And by the way, I didn't get into very scholarly articles. I restricted myself to Wikipedia and such kinds of sources. But you could see a very clear connection between where work in this direction is going and how it might impact sustainability in a variety of ways. Of course, there's a lot of implications on human health and the social side of sustainability, but also on soil fertility, a variety of other aspects that we were actually looking at within the school as part of what we call the food, energy, water nexus. So in that sense, I think while I'm sure there are some very detailed discussions that will happen here at this conference, I think what is being discussed here is also of broader interest to the larger IIT Madras community. And from the School of Sustainability's perspective, we'd be very interested in seeing at some point what linkages we could form with what comes out of this conference and how IIT Madras could support this going forward. Part of what our office does, I'm part of the Office of Alumni and Corporate Relations, is to try to get, as the name would suggest, alumni and corporates to engage better with IIT Madras. I believe we have been able to, in a small way, support some of these initiatives in the past, but we're constantly looking at, I think what is very clear is that if institutions like IIT Madras are to go beyond where we are now, where today we are probably one of the top universities in India, but the aspiration is to be one of the top universities in the world, we are going to do that when alumni and corporates really have meaningful partnerships with the institute. And those will happen only in areas where there is some vibrancy and energy, and this is clearly one such. So again, we are hoping that at some point have deeper discussions with Karthik and others as to what's going on and see how best we can bring in players from outside the academic ecosystem to support not just partly financially, but partly also in terms of, we call this the 3 T's, time, talent, and treasure. So of course, we want people to finance this kind of work, but also provide us with time and talent to help grow these, sometimes shape research questions in directions that are of both scientific and practical interest and so on. So we're very excited to have this here, and I hope you have a great few days at this conference. Thank you.
Thank you, sir. We also call upon Professor Kasturi to say a few words.
Thank you very much for all coming to this conference. And a couple of years back, just after the pandemic, COVID, that we had discussion with Karthik and had that first. For me, that was the first international symposium that we did together. Now I'm seeing that various different people, including the youngsters, are here. So I was with NASA JPL until last few months back. Now I am retired. And a few years back, I committed that I wanted to give back to my country. So I started this space biology program in various institutions, especially Indian Institute of Science is the headquarters. So this is the third year running. I'm running the space biology course, and it's extending to IIT Gandhinagar and also coming back to IIT Madras in 2027. So this is one of its kind to start and show what we have done together. And now Karthik has taken to the next level. Wherever you are seeing there, habitat and space, that is the one that we worked with Karthik, IIT Madras, not Karthik Anandraman, for the past 5 or 6 years, and a lot of young scientists involved and published papers, and it actually generated some sort of good vibe. And I'm here to welcome all of you, and I'm here to get communicated with other Indian institutions, and also I'm going to help ISRO and advise them on the Human Space Flight Center as well. Once again, welcome all of you for this wonderful opportunity, and take it. And a lot of seniors here, and established scientists in the world, and take this opportunity, especially the women here, come forward and talk to them. Okay, thank you very much. You're all welcome here.
Thank you, sir. We'd like to felicitate the deans.
If there are any announcements we can make, we'll start at 10 sharp.
Okay.
We're ahead of time, but we just...
You could post your questions after every session at Slido. The numbers are put up there. And we'll begin in like 5 minutes. An announcement, tomorrow's cultural event at Dakshin Chitra, whoever is interested to join that can please meet at the registration desk and register yourselves for it. Later during the break. Not now. Yeah, speakers are auto-registered, but other participants.
No, nothing. Oh, cool. Let me just see. So there's a laser pointer. Oh, and it works. Fantastic. This is always okay. What about like? Did you get done? Well, I have a pretty soft voice. I don't project my voice very well, so.
I think that's Hannah's microphone.
Oh,
okay.
Just in case someone's not on time. Yeah. Whatever is comfortable.
I actually have a problem once.
Are you introducing?
Yes.
Okay, cool.
You want to for bio, right?
Yeah.
Yes,
So, are you introducing?
I will first
I'm
introduce
going to turn it off
give
for
a
now.
short bio.
Are you giving the bio?
I was going to
Yes,
turn
and
it
then
off.
I'll
So
also like to. So you
And I'll
I want
introduce.
it off.
anything you would like beyond this? I have something beyond this as well.
You can do whatever you want, but just don't make it too long.
I don't know. How to view.
seeing all the 3 days?
Yeah, it's just
You guys are?
Prithvi.
Prithvi and Mazia. Mazia. You're Karthik's boss. Are you also a
With
PhD student?
I'm a postback.
I see.
It's 10 minutes. It's past 10. So to begin the sessions with, we have our opening keynote, which will be delivered by Dr. Gillian Banfield. So this session will be chaired by Dr. Karthik Anantharaman. He's an associate professor and Vilas faculty fellow in the Department of Bacteriology at the University of Wisconsin-Madison, where his lab studies microbial and viral ecology with applications for human and environmental health. Dr. Anantharaman obtained his PhD from the University of Michigan, studying the microbiology of deep-sea hydrothermal vents. He is also the recipient of several awards, including NSF Career, the NIH Outstanding Investigator Award, and the ASM Early Career Award for Environmental Research. I'd like to call him to please introduce the speaker.
Thank you so much for that introduction. I'm here to introduce Dr. Jill Banfield. Before I introduce Jill, I also want to say that a lot of what, she just read, Prithvi just read my biography here essentially. And I will also express my gratitude to Jill because I was actually a postdoctoral student or a postdoctoral scientist in Jill's lab back in the day. Jill is a distinguished professor at the University of California, Berkeley in the Department of Earth and Planetary Science and the Department of Environmental Science, Policy and Management, and Material Science and Engineering. She's also the director of the microbiology program at the Innovative Genomics Institute. Jill's groundbreaking research spans the field of nanogeoscience, microbial ecology, geomicrobiology, and metagenomics. Her work has significantly advanced science in multiple fields, going from material science all the way to microbiology. Of course, you're going to talk to her. Of course, you're going to hear her talk about microbiology. But one thing I want everyone to sort of appreciate is when we think about scientists and when we think about what we are doing, we often go into a comfort zone. We think we are experts in one field. We don't step out of our comfort zones. But one thing I want you all to appreciate from Jill is that Jill's field has gone all the way from mineralogy, geoscience, to microbiology. Jill is a US National Academy member for her contributions in mineralogy, and she's going to talk about microbiology. Her laurels in microbiology speak for themselves. So just appreciate the breadth of research that Jill Banfield has conducted over the course of her career. She has received numerous prestigious awards, including the Anton von Leeuwenhoek Medal, the VM Goldschmidt Award, and fellowships from the Royal Academy and the American Academy of Arts and Sciences. So with that, I'll invite Jill to come.
Good morning. Good morning, everyone. Can you hear me okay? I would like to start by thanking the organizers, particularly Karthik and others, of course, for the opportunity to come here today to talk to you for the cultural as well as the scientific exchange. This is actually my first time in India. So I'm going to talk to you today about some research that I've really been excited about for the last few years. It's a small slice through the kind of work that we do, but it's work that I've been very closely involved in myself, so I have a particular connection to some of the results I'll be sharing with you today. So actually it's now 2025. For 2024 we celebrated for the entire year that it was 20 years since the first publication of a study by Gene Tyson et al. That came out in Nature and was actually the first genomes from metagenomes. So 20 years is a long time ago. Looking back, this was a study where we sampled microbiomes from an extreme environment. It was a system dominated by a few organisms, and it worked. And we used very little sequencing. But people said for many years, it works in acid mine drainage. It will never work anywhere else. Well, I have to tell you that was not a very good prediction, because nowadays people use the method of sampling DNA out of whole communities and reconstructing genomes across every environment you can think of, including the most complicated environments such as soil. Well, a few years ago now, we stopped and we paused and we took stock of the kinds of genomes that had been reconstructed from natural systems. And Laura Haug et al. Published this new view of the tree of life. And it wasn't really intended to be a view of the tree of life so much as it was to be a statement of diversity associated with uncultivated lineages, by which I mean candidate phyla, major groups of the tree of life that lack any isolated representative. And on this tree, you can actually see the individual major groups for which there were no isolated representatives at the time, indicated by red dots. Two things really came clear from looking at this tree, and actually many things, but one of the things, the features that stand out is this group over here, which is comprised entirely of candidate phyla, major groups lacking isolated representatives. And so really just as a description of the feature of the tree, we started to refer to it as the candidate phyla radiation. Also evident is a large number of organism lineages that are associated with the archaea. These are referred to generally as the depan archaea. What's really interesting about these, in addition to the fact that they have no isolated pure culture representatives, is that they have really tiny genomes. And from the genomes, we could predict that there were probably symbionts of other microorganisms. And this is proven to be true and is increasingly well documented in studies from labs around the world. In addition to getting genomes from organisms, there's a lot of other genomes that come to light through genome sequencing and analysis of the data. And I'm going to talk not about microbial genomes today, but about extrachromosomal elements. On this scale, this is megabase scale, we have an indication of the size of these symbiotic CPR bacteria and depan archaea genomes of these microorganisms that are between about 0.55 megabases and sometimes over a megabase in size, so really tiny. Added to this scale, we realize now that there are phages, bacteriophages, that have genomes that actually overlap and exceed the size of these tiny symbiotic microorganisms. A study published now 5 years ago reported many genomes for bacteriophages that were generally in one piece, in some cases curated to completion, the largest of which was 0.74 megabases in size. This can be put into context by looking across the boundary into domain eukaryote, paralleled by the discovery, actually many years ago now, 2023, of the large or giant eukaryotic viruses. This is an example of a Mimi virus, which is perhaps one of the best known giant eukaryotic viruses. Today, we know, 10 years after this first report, that there are now giant eukaryotic viruses that are up to 2.5 megabases in scale. That seems huge, and it dwarfs the size of the bacteriophages reported to date. But let's just step back and put this into context. These viruses of eukaryotes replicate in eukaryotes, and they have really big genomes. So it's a bit like comparing, I don't know, like kiwi fruit and what are those giant fruit you see in India? I forget now. What are they? Jackfruit. Thank you, the jackfruit. We just got introduced to these the other day. Jackfruit. Like really, it's like apples and bananas. So if you look at the size of the some of the giant bacteriophages or phages, they are in one example group about 0.5 megabase in size, with hosts that are about 2-3 megabases in size. So bacteriophages are genome 23% the size of the host genome. So it's actually really huge. Even though these giant eukaryotic viruses are much larger, the hosts are much larger, so they're just only 2 to 4% approximately the size of the hosts. So stepping back and looking at the diversity of these large phages as reported in 2020, a few things come to light. This is a complicated figure, but let me just walk you through it a second. So this is a tree on the interior part that was made from the terminase protein sequence. It's sequences from the study we conducted, and those are in red, and sequences from public data. And perhaps the most important thing to be taking away from this figure is the outside ring, which is indicating the size of the genomes of these bacteriophages. We can see phylogenetic groups across this tree that have been labeled with color, where all of the representative phages have large genomes. We can also see by looking at the environment of origin that these clades of huge phages come from across Earth's environments. Just as an aside, we had quite an international authorship list for this publication, and we let the authors choose the names of these clades, and we decided to use huge phage as the general concept, and we named the various groups based on the language of the authors. So maybe you can find an Indian one in there. Okay, so it would appear that this phenomenon of huge phage genome size has arisen multiple times and been persistent over long evolutionary periods. And to put this into context, these are small genomes. This is a clade of big genomes, small genomes. So it seems like it's a phenomenon that's happened on multiple occasions by one process or another. Another view of this is shown here. We have the genome size on the x-axis and the number of genomes of that size on the y-axis. Now, at this time, the public databases told us the average size of a bacteriophage genome is around 55 kb. Through this study, we had over 200 genomes and 35 curated to completion from metagenome, so we're really confident about the sizes. And that extends the size range like this. But you can see we're still, this is a zoom in, we're still like almost really down at the noise level, but most definitely not noise, extending out the size to a really substantial length. This is the largest genome reported in the study, which was 735 kb. Recently, but unpublished at this point, we're working on genomes now that are up to 860 kb, and who knows where this will end. Maybe some of you have much larger genomes that have not yet been published. But it's a fascinating phenomenon, and we have some indication of the significance of genome size from a few isolated phages that are larger than normal. Phage G is a very famous example with genomes up to about 630 kb. And then we have other groups that are really actually very prevalent in human and animal microbiomes that span a range of genome size here from about 465 to 660 kb. I'm going to step back and ask, what is it about these genomes? What can we learn by looking at the genomes? What interesting phenomena come to light? And I'm going to focus first on this study by Audrey Devoto et al., published a few years ago now, that was really centered on this particular clade of huge phages, which we called LAC. Perhaps the most striking thing about the LAC phages, other than their huge, 0.5 megabase scale genomes, is that they use an alternative genetic code. And I think this is actually really still rather underappreciated as a phenomenon in bacteriophages. How do we know this? Well, we can predict the genes using standard methods, and we should expect the genome coding density to be about 90%. So 90% of the genome should encode open reading frames. But when we predicted the open reading frames on these LAC phages, the genomes looked like this. Clearly something's wrong. There's way too much intergenic space. And if you look at where the proteins are terminating, here, 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 and here, where it would seem that the protein should extend on, we find that there's a UAG or TAG stop codon. By analysis of this and by prediction that the UAG is not actually being read as a stop, we can recover the entire protein or gene. And in fact, this reunites fragmented genes to produce genes that are full length and identifiable, for example, as a DNA polymerase. If we make an alignment of the sequences of these LAC phages, which have been recovered from quite a large number of human animal microbiomes, including Indian children actually, align the sequences, we can see what the stop codon is coded for in other related phages. And we find consistently that it's glutamine Q. This has actually since been established by proteomics, not for these phages, but for other phages that use this stop codon reassignment. It's really important to know about this reassignment because if you don't repredict the genes with the proper code, you'll miss a lot of the proteins. You won't be able to work out that it's a phage. And most importantly, these stop codons are especially prevalent in the structural proteins that we look for to say something's a phage. And so this is another complicated tree, a bit similar to the one I showed you in the previous slide. This is a phylogenetic tree in the middle. Identified within this phylogenetic tree, again, are clades that have alternate coded phages within them. So this is a phage tree. What I want you to notice is that the size is highly variable. It's not a pattern in which alternative coding only occurs in large phage genomes. It can be in any size. In fact, it even occurs in prophages integrated in normally coded genomes. And in fact, these phages all replicate in genomes that use the standard code. The other thing I want you to notice from this is that there are TGA and TAG codon reassignments. If you remember, there's 3 stop codons, TAA, TGA, TAG. Some phages reassign TGA, some reassign TAG. But what's most interesting of all is only a little bit evident from this area and this area, for example, on this tree, but I'm going to expand and make this clear in the next slide. But if we read the code here, you can see the reassignment is indicated by a color and the standard code by gray. And what was really interesting to us is that there's very fine intermixing of standard code and alternatively coded phages. And in fact, alternatively coded phages that are reassigned one or the other of the stop codons. So this next table, maybe it's a little bit easier to digest. We have the various clades indicated in the prior tree with colors and names. and we have an indication of what that clade contains in terms of the genetic code. And you can see here several clades have a mixture of standard code, TAG recoded, and TGA recoded. I wanted to mention more or less as an aside that these alternatively coded phages are particularly prevalent in human microbiome, and in fact probably represent about 6% based on our analysis to date, 6% of all phages in the human microbiome. But I think what's really interesting, and perhaps most interesting of all, is this phenomenon of intermixing in clades indicates to us that it's actually pretty easy to switch the genetic code. It's not stable, immutable as once was thought, but actually it's possible to rapidly transition from the standard code to one or another of the alternate genetic codes. And so there are multiple origins of alternate coding in really divergent phage groups. We don't really know why, to be honest with you. And it's an interesting phenomenon, and I'm waiting for someone to correct me on this, and maybe some of you know something that contradicts what I'm going to say next, but to my knowledge, nobody has ever isolated an alternatively coded phage, and I wish they would, because we would love to be able to do experiments with them. And I've said this to many people who are phage researchers, and I think they're looking, but to my knowledge, we have no experimental system. But from the genomes, we can hypothesize how this might work. And the scenario we anticipate is the phage injects its genome into the host cell. The initially injected genome has to make use of a ribosome that works with the standard code. It reads the standard code. So all the early transcribed and translated genes and proteins use the standard code. They don't use the stop codon that's been reassigned. And so those products are fine. Should any expression of the genes that are late and have many stop codons reassigned within them occur, those proteins would be non-functional. And we think that that might be actually really important. So stepping through this, at some point, the phage genome is being transcribed, and a suppressor tRNA will be produced. Now, a suppressor tRNA is a tRNA that reads a stop codon as an amino acid. And so in this case, TAG is read as glutamine. This is something that has to be evolved or acquired. But once it's available, it can start to translate the proteins appropriately when they use an alternate genetic code. So those tRNAs get amino acylated, and it's possible now to produce the late stage proteins. One of the ways that phages respond to, one of the ways that hosts respond to infection is to induce early production of lytic genes that would rupture the cell. That would be devastating to the host. Devastating to the host and also to the phage, but the phage doesn't want that to happen, so preventing early production of lysis genes may be part of the explanation why alternative coding is advantageous. Okay, so big genomes. What's encoded in these genomes? By and large, we don't know because so many of the proteins have no known function. There's some very interesting work done by a couple of different groups, I know specifically in the US, by Joe Pogliani's group. For example, on a phage that is a jumbo phage, it's over 200 kb, in fact, over 300 kb, and it's a Pseudomonas jumbo phage. And I just mention this as an example of what kind of cool stuff has been discovered once we start to explore these huge phages. These phages make a nucleus inside of their host cell. Again, this is not my work, this is work of others. This allows the phage to replicate its genome away from all of the machinery of the host bacterial cell. So the CRISPR-Cas system can't get in and attack the phage genome because it's separated by this phage nucleus. This completely separates the ribosomes from the machinery dealing with nucleic acids. And so the capsid gets assembled exterior to the nucleus, and then it gets packaged at this nucleus membrane with the genome to make mature particles. So just incredibly cool, interesting biology, something maybe you couldn't even imagine without this access to these large genomes, and especially experimentally tractable ones. What else? Well, the thing that came to our attention looking at some of these large genomes was that they had a lot of genes involved in translation. So this is generally large phages. Some of them have lots of genes involved in translation, and in fact, they have genes involved in the 3 stages: initiation, elongation, and termination. Now, this is a cartoon in which the blue represents the ribosome here, and these are obviously bacterially coded. So bacterial coded components are in blue, phage encoded components are in red. what you will see from this is that we find within these huge genomes of bacteriophage components that are involved in all 3 stages, including, for example, ribosomal proteins, tRNA synthetases, and release factors. So it would appear that this is a different strategy to alternate coding. These phages are taking over the functionality of the ribosome, not by switching the code, but by preferentially translating the phage transcripts. So a lot needs to be learned about how this works in detail, but it would seem that this has been a very advantageous strategy. In fact, not just for bacteriophages, but also giant eukaryotic viruses. In fact, when they were first discovered, one of the things people were most excited about is they had genes for translation. So they broke down this phage versus living organism barrier to some extent insofar as they had these proteins which were not expected in extrachromosomal elements. Well, bacteriophages have them too, in some cases many of them. Not all of them shown here, but many of them. So moving on, I want to not introduce metagenomics, but introduce a manifestation of metagenomics that we're really excited about at the moment and that will be central to what I'm going to present for the rest of the talk. So we all know about metagenomics. You extract the DNA, you sequence it, and you reconstruct genomes. And a lot of people most people just accept those genomes. They're draft genomes. They may be contaminated. They may be incomplete. But this is increasingly not necessary. So if we, for a long time, really from the beginning, have curated our genomes, ideally to completion, and sometimes using only Illumina reads, so that's a very challenging thing. Now we're in the long read era. Now those reads are not perfect, and it's not straightforward still, but it is possible to combine, in my opinion, to combine the short reads, Illumina reads, with the long reads to get completely accurate sequences. In other words, curation to completion. And of course, we can do all sorts of things with completely curated genomes. We can make phylogenetic trees with the genes. We can analyze the genome structure. I'm not going to go into this. And we can predict the proteins. But of course, the problem with these extrachromosomal elements is the proteins are very difficult to deduce a function for. And so one of the things I'm going to talk about in the presentation today is the use of in silico structure prediction applied directly to the genomes of extrachromosomal elements to get clues as to what the functions are. And I think this is really a powerful method, especially done at scale across entire genomes, and is really starting to uncover functionalities that remained completely unclear until this point. And I'm going to talk about archaeal extrachromosomal elements, because I really love archaea, and they are, in fact, the group that has the extrachromosomal elements that are least well understood. And this is a little out of date now, but you can see here that relatively few viruses and plasmids have been reported overall for archaea. And they tend to be for the taxonomically well-known groups. So what appeared in our data was very, very interesting and really caught our attention were these genomes, very large genomes, manually curated to completion, that were clearly of extrachromosomal elements that were up to 1.1 megabases in length, but not bacterial, associated with archaea. And they have a very distinct genome structure. First of all, they're linear, terminated by long kilobase, few kilobase scale, inverted repeats, not circular, linear. And almost all the genes are encoded on one strand, on the large replicor. There's a second small replicor. It has genes on the other strand. So it's not, the genes aren't switching backwards and forwards. They have very consistent coding, switching at the terminus of replication. They also feature very fascinating tandem direct repeats, like starters in the genome. And I'm not going to talk about that, though it's something we've looked into quite a bit, and actually is really a marker for these very enigmatic genomes. They do not encode the genes for ribosomal RNAs. They do not encode the genes for things we'd expect to be necessary for us to classify these as organisms. They are not organisms. So clearly not microbial genomes. We deduced that they were clearly not plasmids, or they were not clearly plasmids. There's a subtle difference. And there was little evidence at the time we first reported these things for them being viruses. They have some very interesting features. One of these is the fact that they have low GC content, but then we'll have short regions of the genome corresponding to a cluster of genes that have much higher GC content. And that GC content matches the GC content of coexisting archaeal organisms that we believe are the hosts for many reasons, one of which is very high sequence similarity between the genes of these extrachromosomal elements and the genes or if you're comparing it, the proteins of the putative host. So this really ties these extrachromosomal elements to archaea that are called ANMI2D or methanoperidins, and they are organisms that are very interesting because they anaerobically oxidize methane, which really brings into focus the relevance of these extrachromosomal elements from the perspective of the methane cycle. So we called them Borgs because they weren't plasmids and they weren't viruses. As far as we could tell, they were clearly acquiring or assimilating, if you recognize that term, from Star Trek, Borgs from Star Trek, assimilating sequences from a variety of different organisms. And when we published this paper, the Nature Art Group did a spectacular job of developing a cover to really feature this concept, and I love it. I think they did a fantastic job. But you'd have to be a Star Trek fan probably to make sense of this. Who knows what Star Trek is? Oh, good for you. Excellent. Well, if you only know a few things about Star Trek, it's probably this person, 7 of 9, who was a Borg. And one of the things that was most fun about this study is when one of my PhD students, Luis actually, tweeted about this person whose name seems to have been removed from this slide for some reason, Terry Ryan, retweeted the post. So Terry Ryan, 7 of 9, repeated, oh, this story about Borg. So we're pretty thrilled by that. So what are these genes with this deviated GC content? In this case, there are very interesting and unexpected genes to be found on an extrachromosomal element. These 4 genes encode methyl coenzyme M reductase. And you've probably heard of this complex. This is the complex central to methane production. And in the case of methanoperidins, methane oxidation anaerobically by reverse methanogenesis. And these other 2 featured here encode cofactor 430, which is the cofactor for MCR. So these giant extrachromosomal elements, Borgs, are carrying around genes that are really central to methane cycling. So where are they? Where are they found? Well, they're found in terrestrial environments to date, and a variety of terrestrial environments. Not surprising because of their link to the methane cycle. They're found in anaerobic environments like wetlands. We found them in data sets of others from the Spruce Project in peat. We found them under rivers, in mine discharge. I'm not going to talk particularly about this. This is a rice paddy. And again, this is all, I guess you can read it here. This picture here reflects the fact that we found them in municipal landfills. This is data of Laura Hug. And these sites are obviously very famous for their methane emissions, as are wetlands and rice paddies. So they appear to be in a variety of different environments, and I would say potentially really important from the perspective of the methane cycle, not just the organisms, but the extrachromosomal elements of them. This is a diagram from a view by Thor et al., 2019. It's a cross-section. You can see the air-water interface. It's a lot of information here. I really just want to draw your attention to the fact that we have organic biomass being converted to methane by methanogens, and then we have MCR running in reverse to oxidize the methane. So really a feedback to methane emissions from these kinds of environments. This is an overview of one Borg, just to give you a sense of the fantastic things these extrachromosomal elements encode in their genomes. And that looks like a cell cartoon. And yes, we show a cell, but the only thing shown in this diagram are the genes associated with the Borgs. We've divided this into 3 sections, and I'm going to start down here that these Borgs carry a lot of genes involved in stress response. They encode pathways for PHA, polyhydroxybutanoate, production of carbon storage compounds, suggesting that they're enabling their host organism to survive under a wider range of conditions than may otherwise be possible. They contain a very interesting and weird set of genes involved in central carbon metabolism. But most important, I think, they encode genes involved in energy metabolism. Not all of them, by far, have MCR, but this is one case that does. Ferredoxins, carbon monoxide dehydrogenase, and a wonderful, extraordinary set of multi-heme cytochromes. Who ever heard of multi-heme cytochromes on extrachromosomal element genomes? Well, not just one, but dozens of multi-heme cytochromes, sometimes having many, many, many heme-binding motifs, as indicated by these little boxes. Some associated with the cell membrane, some with the periplasm, sort of that kind of space, and then some extracellular. working with a collaborator who's an expert in multi-heme cytochromes, and specifically the multi-heme cytochromes that assemble into nanoeyes. We deduce that the multi-heme cytochromes encoded on these Borg extra chromosomal element genomes provide the proteins that you need to make nanoeyes. Why do you need nanoeyes? You're oxidizing methane. That releases electrons. What are you going to do with the electrons? You need to get rid of them. In this case, these organisms want to deposit electrons onto iron oxide minerals, or manganese oxides, as shown by others. That means they need to get the electrons from the interior of the cell all the way out into the tiny spaces in the soil where there are iron oxide minerals available. And so it would appear that these extra chromosomal elements provide additional capacity that enables methane oxidation to go to completion. We have some transcript data from soil. That's really hard to get, but some transcript data from one of the Borgs, and we find actually very high expression of these multi-heme cytochrome proteins that are very, very likely able to assemble into nanoeyes. Okay, I'm just going to skip over this, I think, very briefly. There are other types of Borg. Everybody always asks me, are there other kinds? Yes, there are. This is a Borg. We have mini Borgs. There are some that are really tiny. They share all of the features, but they just simply have much smaller genome sizes and less interesting repertoires of proteins. So moving on, methanoparatins has a variety of extra chromosomal elements associated with it. We've used it as a kind of case study for archaea, for which we can document their associated extra chromosomal elements. This is methanoparatins here. This is a tree made on the overall proteomes of these extra chromosomal elements and their hosts. You will see the host genomes. They've got plasmids and viruses we can now readily identify that associate with them. There's the Borgs and the mini Borgs. And so it would appear that methanoparatins is associated with a constellation of extra chromosomal elements of all different types. And there's evidence for a pretty extensive lateral transfer of genes between them. So really very, very important associations, I would predict, between the host and the extra chromosomal elements in terms of evolution, in terms of protein innovation. So what about these Borgs? Well, one of the things that really came to light that surprised us is the copy number, the apparent copy number. It would appear that these Borgs exist in a ratio not uncommonly of 8:1 in the host cell. Now, to put this into perspective, this is more or less to scale. This is the methanoparatins chromosome. These would be the Borg genomes. Should there be 8 copies per cell? So what is going on here? What are they? Are they more like parasites, or are they partners? If they're like viruses, maybe we can find clues to viral-like behavior from the genes and their defense systems, the avoidance of host defense systems. If they're like plasmids, which a lot of people concluded pretty much immediately they probably were, they should have genes that assist the host in surviving under a variety of conditions, such as the ones I just explained. So how do we get beyond the situation where we are now, we were at that time? I decided that one of the things that would be really interesting to do over my sabbatical was to actually prioritize analysis of the multi-copy proteins. So some proteins, some functions occur in one copy in the genome, and some are in multi-copy. So we assigned all the proteins to protein subfamilies, which is just basically one kind of protein, one kind of function. And then we asked, what is particularly highly represented in the genomes? What occurs in multi-copy? And surely, if it's in multi-copy, it's got to be important. So let's focus on those. To put this into context, only about a quarter of the proteins even had PFAM annotations. So the way we decided to move forward from this was to make use of these structure prediction tools with the expectation that these are highly divergent proteins relative to the biochemically characterized versions, and that although the sequence similarity is not conserved enough for us to recognize it, the structure may be conserved. And so that's what I'm going to talk about pretty much for the rest of the presentation today. We have now at least 17 genomes, of which 13 are curated to completion, and some have actually been validated by long read. So there's actually really very well-established genome structure and size, the largest being about 1.1 megabases in length. Across these 17 genomes, there's about 20,000 proteins, and we've predicted the structures of all of them. I'm not going to present anything like that. I'm just going to pick out a few really interesting examples that provide some kind of insight into what these Borgs might be up to. So this is a summary of the most abundant, highly represented subfamilies of the proteins that are in multi-copy, colored by the Borg type. We name the Borgs after colors. So there's a ruby Borg and an amber Borg and a green Borg. And this is on the y-axis is the number of proteins in that category. One of the things that we realized by structural analysis pretty much immediately is that sometimes these subfamilies are not perfect. And in this case, 2 of the 3 most highly represented subfamilies in these Borg genomes appear to have the same function, the same structure. They were just assigned to different subfamilies. And a couple of less abundant subfamilies combined also have this structure, and they're indicated here. And if we combine these and ask how many copies of this protein type are there in Borg genomes, the average is 18 copies per genome. We construct a phylogenetic tree for these. We can see that these sequences are in fact intermixed, again, supporting us combining these into a single functional type. So what are they? Well, very few, if any, had any significant hits by any kind of annotation method, and many of them, even with structure prediction analysis methods, the structures were fine, but they didn't have any close similarity to any known characterized protein. A few had low scores to these things called AMSH proteins, which, if you work on eukaryotes, you may recognize, but they're basically zinc metalloproteases. I was really concerned, actually, when we started to use AlphaFold 2, that we had these novel proteins and we have these templates based on the protein database, things we knew, and we were just kind of forcing these proteins to fold up such that they would match something that was in the databases. So one of the checks we did early on is to take out the templates from the protein database, just get rid of all the known structures and just calculate the structure based on the sequence alignment of the 315 Borg proteins. And to my delight, actually, it recapitulates the predicted structure using the PDD templates very well. So this AlphaFold really actually works very well. You can see the confidence in blue, so the high confidence is in blue, indicating we really probably have approximated the structure of these proteins. So what is AMSH? They're proteins best known in the eukaryotic world, and they remove the protein, a small protein called ubiquitin, from proteins that have been labeled with ubiquitin, probably to send them to the proteasome for destruction. You can see here an example from the public data. This is ubiquitin, the label. This is the protein, AMSH protein, that actually cleaves ubiquitin off this labeled protein, rescuing it from destruction by the proteasome. And if we look at the active site of some of the examples from the Borgs, and just a few shown here, and the reference structure, they have exactly the expected residues we would expect in the expected places, such that they would bind zinc consistent with their function as zinc metalloproteases. If we take the sequence alignment from the 315 proteins from the Borgs and we map the sequence alignment showing where the conserved regions are onto the protein structure, where conservation is red, we can see actually this really brings to light the active site. The conserved regions are this his, asp, and then the serine, which is necessary and expected from zinc metalloprotease. So this is really useful generally for localizing the active site of an unknown protein. But archaea really are not expected, at least for the most part, to use ubiquitin. In fact, they use a similar kind of label called SAMP. And they sampleate, proteins are sampleated much for the same purpose as proteins are ubiquitinated. So where did the SAMP come from? When we look at the genome of the host organism, we can find the gene encoding the SAMP, the small archaeal modifier protein, the ubiquitin-like protein. And we can find the structure, of course we have the structure, of the D-ubiquitin ases, in this case desamplases, and we can simultaneously predict how these structures interact with AlphaFold. So this is basically take this protein, and just see whether or not they interact. And in fact, that calculation predicts that the GG tail of the SAMP binds exactly into the active site of the desamplase. Well, who adds SAMP to the proteins? The Borgs do not have that capacity, but the host does. So the host has an E1 SAMP adding protein, and when we do the same experiment in silico, the SAMP from the genome docks perfectly into the sampylation enzyme from the host. So you might think, okay, well, these are a lot of proteins that have just been randomly generated and duplicated in the genome. They have no functional significance. But we actually have transcriptomic data from in situ for one particular Borg called Black. And Black Borg has 24 different desamplase, putative desamplases in its genome. We have nanopore, so these are long-read metatranscriptomes, and you can see we have evidence in these various samples for simultaneous expression of these proteins, suggesting that they are actually active and used in situ. So why would you have up to, the maximum, 32 copies of a desamplase in a Borg genome? If we look at the predicted structure of these different desamplases from the same genome, this is, in this case, orange Borg, this is just 4 examples, you can see the SAMP is bound to the desamplase. The protein that would be labeled and from which the SAMP would be removed would dock into here. And we can see that the volumes are really rather different, suggesting that these desamplases, 32, up to 32 per genome, on average 18, recognize different sampulated proteins and desamplate them. So what are they doing? They're rescuing Borg proteins that have probably been targeted for destruction by the host by cleaving off this label specifically. So of course, this is a hypothesis. It's not been tested, but it's the interpretation that we would present for future experimental work. There's a lot of other really interesting things that these Borgs encode, including a lot of genes for production of molecules, like glycoconjugates molecules that decorate the cell surface, put simply. And this is just one region of one genome. You can see there's dozens of genes that have functions related to the cell surface or the cell wall, and many of these are occurring in multi-copy. I'm just going to give you one example of one particular prevalent protein encoding in the Borg genomes, which is called diolycyl phosphate mannose synthetase. And that's a mouthful, so I'm just going to call them DPMS. We're going to take a little bit of a look at these proteins, which basically involved in glycosylation of surface proteins. So we can predict the structures, and we can compare them to the structures of known compounds. In this case, it's an almost perfect match, the gold being the Borg protein, the blue being the reference structure. And in fact, the active site is exactly as we would expect it, across many residues, more than shown here. What's really interesting in these multi-copy proteins is that we have a variety of different conformations of the protein. In fact, some completely lack this membrane anchor. They just have the cytoplasmic domain. And we have reason to believe that would still be functional based on some experimental evidence for the reference structures. And some of them, lo and behold, have double cytoplasmic domains. So we don't really understand what they might be doing, but they're obviously conferring structural variety to these seemingly really important proteins involved in glycoconjugate production. And I'm going to flag in the next slide those that have this extra cytoplasmic domain. And just look at the phylogeny of these. So this is a tree made from this DPMS protein sequences. And in this tree, the Borg sequences are in black, and the methanoperidine sequences, because they have 1 or 2 per genome, are shown in blue. And I really only want to focus on one thing in this tree, and that is as follows. Some clades, this one expanded here, we find a huge variety of archaea that have this protein encoded in their genomes, and they cluster with the Borg proteins, so many, many, many proteins from ANMI, from methanogens and other archaea, indicating almost certainly that the Borgs acquired this DPMS from their hosts. So transfer from the hosts to the Borgs. But what's really interesting is we look at some of these other clades, for example over here, the vast majority of sequences are from Borgs, and there's just be one popped in there from the methanoperidines. And actually this is the kind of first evidence that the proteins are going back from the Borgs to the hosts, suggesting that they may be basically serving as incubators for sequence exploration. So this is showing the same thing in a slightly different way. This is the DSAMPs. This is the DPMS. You can see here pretty clear evidence of transfer from the hosts to the Borgs. And in this DPMS tree, you can see evidence for transfer from the hosts to the Borgs. And then this is the expanded part that shows the opposite. One of the most interesting things that came to light through structure prediction, quite really unexpected, was the indication that Borgs make capsid proteins. Now remember, everybody's saying they're plasmids, right? All people who reviewed the paper, who saw the paper on bioarchive, their plasmids, of course, their plasmids. Well, wait a second. Some of the proteins fold up exactly as we'd expect if they're capsid proteins, showing very strong structural similarity to capsid proteins of viruses of archaea, small viruses of archaea. And what's rather interesting is we take these proteins and we multimerize them, we find that they multimerize into hexamers with very high confidence. No other conformation other than 6-mers will give us these kind of scores, suggesting that they form these arrays. And in fact, if we put up to the limit that we can use with AlphaFold 3, 24 of these proteins in and we co-fold them, we can, on some occasions, it's not entirely reproducible for reasons to do with the software tool, but we can actually show that these hexamers assemble into sheets. Very consistent with the fact that they're making a protein coating for something that might be encapsulated in the Borgs. The other thing that comes to light looking at the transcript data is that 75% of the transcripts of the Borgs localize to one region of the Borg genome, as shown here. And this last gene is a capsid protein. So these proteins are very highly expressed, and that's exactly what we'd expect for a capsid of a virus. So what are these other proteins that are highly expressed? We don't know, despite all of the efforts to explain or predict their function. We still don't know what those highly expressed proteins upstream from the capsid protein might do, but nonetheless, we can find that they're highly conserved. So these capsid-like proteins are present in all Borgs and in the mini-Borgs, and the context is relatively conserved. So we have very strong evidence, I think, that these are really quite important and I think suggest pretty strongly that they are, in fact, something to do with encapsulation. Stepping back a bit, we can look at many of the other functions that come to light through a combination of approaches, especially including structural prediction, and there's a list down here. If you run your eye over it, you can see energy, translation, glycolysis, TCA cycle, nucleotide synthesis, coenzymes, and so on and so forth. If we look at the presence-absence patterns in the genomes, these are the Borg genomes as columns, we can find it's kind of patchy, so some have them and some don't. But what's really striking is that the color scale indicates the position in the genome. Are they early in the genome, middle of the genome, late in the genome, as indicated here? Where the proteins are present, they have conserved location, generally speaking. So if the protein occurs in 6 Borgs, it'll always be early, or it'll always be middle, always be late. What also comes to light from an analysis of this kind of list is the very strong parallel with giant eukaryotic viruses. This is a list of some of the functions that have been reported in giant eukaryotic viruses. Remember mimivirus, for example. Very parallel, very similar kinds of protein functions. It's also true, at least from the perspective of some of these systems, that they are prevalent in large phages, the viruses of the domain of life bacteria. So, parallels between Borgs and giant eukaryotic viruses. Borgs have linear genomes terminated by long inverted repeats. Many giant eukaryotic viruses have that exact genome structure. They have lower GC content. They have some similarities in genome organization. Giant eukaryotic viruses also have proteins involved in ubiquitin metabolism. Also, they share the capacity to apparently make glycoconjugates, possibly for cell, host, or capsid surface decoration. They take up genes from their hosts, as is well known for giant eukaryotic viruses. Their genomes feature a lot of gene duplication, repeated functions, and their gene inventories are rich in functions we don't expect to find on extrachromosomal elements, such as the long list I just showed you previously. So, despite the conclusion of others that almost certainly these are linear megaplasmids, we think they probably are more likely to be viral-like elements. They join giant eukaryotic viruses, in other words, giant viruses of domain eukarya, with giant phages, the giant viruses of bacteria, and thus maybe are giant viruses and smaller versions of domain archaea. So, in terms of their characteristics, their extraordinary metabolic capacities, not related to replication, not related to defense against host immune systems for the most part, might indicate that they long-term associate with their hosts. And counter to that, They have putative structural proteins, such as capsid proteins, sometimes extremely high ratios of abundance compared to the host, up to 50 to 1, suggesting that they encapsulate and exist outside cells, suggesting that they may be more lytic virus-like. One of the things that we realized in the 2024 study by Marie Shaw-Merrick et al. Is that if we look at the positions of genes that occur in one and only one copy in every genome, so they're completely conserved across the Borgs, we find that their positions are consistent across the genomes, here organized by length of the large replicome. Based on the more recent work that I've been doing over the last couple of years, I would expand this to say that 64% of all the genes approximately share the same position on average within plus or minus 10% of the genome. So conservation across these genomes with a lot of evidence for gene loss in some of the Borg representatives, as well as, of course, a lot of gene gain. So concluding thoughts, where did they come from? This is obviously an interesting topic. There are competing theories for giant eukaryotic viruses that also probably apply to these Borgs. One of the ideas is that these used to be an organism, and the organism underwent genome reduction and loss of function and ultimately became a giant virus. The opposite idea is that they undergo genome expansion by acquisition and organization of their genomes, probably more paralleling the giantism in bacteriophages, which I showed you as something that's arisen many times and has arisen many times in giant eukaryotic viruses. So we don't think that they have a common ancestor with giant eukaryotic viruses, because I didn't go into this, but the protein phylogenies don't support that. So we think that they have no common ancestor with giant eukaryotic viruses, suggesting they have independent origins, probably acquiring these characteristics through convergent evolution. So in other words, if something's going to become giant in one domain of life or another, the same kinds of functionalities are advantageous to acquire. I want to turn attention back to the mention of the nucleus-forming phages, which I talked about from the work of others, and mention briefly the fact that giant eukaryotic viruses sometimes make nuclei inside of a host cell, a host eukaryotic cell. So both bacteriophages and giant eukaryotic viruses do this. They make a nucleus. And so this, I think, is extremely interesting from the perspective of evolution. And I'm just going to skip to this last point because it's perhaps the most interesting thing going forward. It has been suggested by some that the origin of the nucleus in eukaryotic cells was actually viral. The viruses can make these structures that encapsulate the nucleic acids and separate them from the cytoplasm. It occurs in eukaryotes, eukaryotic viruses. It occurs in bacteriophages. It may occur in Borgs. Is it possible that giant viruses or viruses of some kind were actually the origin of the nucleus, but also the origin of the nuclear genome? So if that's true, and if by chance that the Borgs and other giant viruses of domain Archaea became the nuclear genome, then we're all basically Borgs, which I think is something to end on. Of course, this remains very speculative at this point. There's no proof of this, but there's a lot of reasons why I think this idea of a viral origin of eukaryotic nucleus and nuclear genome actually has a lot of traction. It remains to be seen. We're actively collecting large genomes from bacteriophages, from viruses of giant eukaryotic viruses, and of course, trying to find more examples of giant viruses of Archaea so that we can look for the kinds of functionalities that might explain or contribute to the missing pieces of the story of the origin of eukaryotes. So with that, I'm going to conclude. I hope I didn't talk too long. These are some of the co-authors and collaborators that have contributed to this work. Borg people, alternatively coded phage people, a bunch of other collaborators. This is work that I'm particularly invested in. It's my sabbatical study that's gone on and on. You can find a preprint on bioRxiv if you'd like to read more about it, and I would just like to thank you all for listening to me. I'd be very excited to talk to you about your thoughts on this. It's been very beneficial for me to meet and talk with people at prior meetings who work on this huge collection of viruses across the 3 domains of life, and I hope that this will be interesting. Okay, thank you, and if I have time for questions, I'd be happy to take them.
So we have a few questions. We have a few questions up here, but are there any from the audience?
Thanks very much, Joe. That was great, as always. I had a question in relation to the inverted terminal repeats. Are they in any way, shape, or form similar to telomeres? And in relation to the Borgs, do you see any genes that would suggest that they are also nucleus forming?
Yes, this has been suggested in the first publication that they may have genes that implicated in nucleus formation that hasn't been explored beyond that. And everybody asks me about telomeres, and it's a very interesting question. I think that there is indication that they may have telomerase-like genes. But it's not, again, it's still not certain, but it's something that we suspect very strongly. Yeah.
I'm curious if you've looked for like some of the Philip Bell like ideas like mRNA capping and things in Borgs or other places.
mRNA capping enzymes, I don't think, yes, and of course that's Borges, the person who's most well known for his ideas about phage viruses and eukaryogenesis. I don't think we found mRNA capping enzymes. I think that's something that we've definitely looked for and have not come to light. But they do actually have amazing things like escort systems very clearly, and they have histones, histone-like proteins, and a lot of other things that we might be pointing to in that regard. And I didn't have time to talk about that today.
You have a question here?
Yeah.
Brilliant talk. I was wondering, two related questions. Is there any variation across the different copies of the Borgs within single cells or between different cells of the same strain or species of the host? And
Yeah,
what's the replication machinery by which you get these multi-copy Borgs in the same cells?
Yeah, so the question of the host associations is a particularly difficult one because we're dealing with samples that have many, many Borgs and many, many hosts. So we don't have a good, strong host-Borg linkage. In some cases, we have circumstantial evidence where there is actually only one Borg and one host, like black Borg. So we think that they probably do have specific hosts. The other thing we have is evidence of recombination between some of the more closely related Borgs, suggesting that they do sometimes co-localize in the same cell. And I think there was another part of your question I wanted to answer, but I forget. Oh, how do they replicate? We only know about how they replicate from the perspective, well, they have a lot of machinery for replication, but we only know about the replication style from the GC skew of the genomes, and that would indicate they're replicating from the termini to, sorry, from the terminal repeats into the terminus replication at the junction of the two replicors. And that's all I can tell you, I'm afraid.
Hi, over here.
Oh, hey.
Excellent talk, Jill. My question is related to that of Paul. I saw that you annotated in the Borgs some major vault proteins. Do you think those
Thank you. Thanks.
Hello, check, check. Tea break folio.
podium to put it.
என்னாச்சு இது இன்புட்டே வரல I see.
கனெக்ட் பண்ணுவா அவர் கனெக்ட் பண்ணல
We now move into our first session, Fudge in Microbiomes. This is going to be chaired by Professor Kasturi Venkateshwaran and Dr. Sudarshan Govindarajan. I'd just like to give an introduction to our session chairs. Professor Kasturi has over 47 years of experience in marine food and environmental microbiology. He has made significant contributions to advancing the understanding of microbial life across diverse systems. And as a lead at the ISS Microbial Observatory projects, he has spearheaded the exploration of microorganisms inhabiting the US nodes and the Kibo-Japanese experimental module. His work has been instrumental in revealing microbial resilience and adaptability, shaping the foundation for future space missions and deepening our knowledge of life's potential in most inhospitable environments. Our other session chair is Dr. Sudarshan Govindarajan, and he's an assistant professor and DBT Wellcome Trust Early Career Fellow at the Department of Biological Sciences at SRM University, India. His research focuses on jumbo bacteriophages, bacterial defense systems, and developing alternative approaches to combat antimicrobial resistance.
I should not get introduced more, than Karthik. He's going to be the speaker here, and we all know this Karthik Square, and he's the second one in this conference. And I came to know about him when we are convening this particular conference this time. And he's an associate professor now in the University of Wisconsin-Madison. And before that, he had his postdoctoral fellow with Jill Bansfield's lab. And before that, he had his BTech in National Institute of Technology, Karnataka. And he's also a recipient of several awards, including NSF Career, the NIH Outstanding Investigator Award, and the ASM Early Career Award for Environmental Research. His interdisciplinary research program was a combination of computational, laboratory, and field-based experiments to develop and apply novel approaches to study the viral diversity, ecology, and evolution in environments ranging from soil, lake, ocean, and the human gut. So here is Karthik. Thank you.
Thank you so much, Venkat. Okay, I think now this is probably better. So I'm delighted to share some of our work. This is long-term work that I'm sharing. Before I sort of start, in the course of the introduction of my talk, I will talk about everything our lab does. I have some students from our group presenting posters as well that I'd encourage you to check out. But what you're seeing over here is a view of Lake Mendota. I just flew a drone over it, and that's essentially what you're seeing. Lake Mendota is a beautiful lake, and our University of Wisconsin campus sits right on it. This is essentially our building, Microbial Sciences. So we can just get down to the building, take a boat, and go out into the lake and sample. And that's essentially what I will talk to you about today. That is something my students have enjoyed doing. So as Venkat introduced, I am an associate professor at the University of Wisconsin. But what also seems to be lost on here at the bottom is that I'm on sabbatical at IIT Madras. I've been here for a year. I've been here for 6 months. I'm going to be here for another 6 months. So I'm here for an entire year. So my laboratory is quite diverse. We are about 50% computational is what I'd like to believe. We are a little more computational than that. About 50% of our lab essentially does bioinformatics and what I broadly call phage biotechnology. So we develop software, we also think about applications of phage in different sort of settings, as well as viruses, not just phage, but we also think broadly about viruses. The remaining 50% of our lab is very broad ecology and evolution focused. We have active projects in human microbiome, so looking at diseases, looking at the response of the microbiome to different sorts of gut disorders, including colorectal cancer. And then we are interested in freshwater microbiomes, marine microbiomes, and soil microbiomes. And an emerging common theme in all of our research is the real power of longitudinal metagenomics or time series metagenomics to help study ecology and evolution in nature. So this is something we are pursuing in freshwater, in marine systems, and soil. So as far as marine and soil is concerned, I have students who are presenting, and I'll tell you a little bit about some of our freshwater work that we're conducting. Whenever we enter any environment or try to study a new question, we often wonder, where do we start? And everything in our laboratory starts with a very simple question. We are interested in understanding the roles of viruses and phage. And then we can tack on the ecosystem, the environment, and everything on top of it. So now, for example, we are interested in studying the roles of viruses and phage in modulating either environmental or human health. Now, where do we start? So we look at everything through 3 different lenses. So we look at things through the lens of diversity. So for example, we can ask the question, how can we accurately identify and characterize viruses in microbiome data? We can look at the lens of distribution. So we can study viruses across some sort of a transect, so spatial transect, temporal transect. So we can ask questions like, can we use viruses as predictive markers of diseases or even environmental health? And finally, we can look at questions through an ecology lens. So we can ask questions like, what are the roles of viruses in either, let's say, driving gut dysbiosis, nutrient cycling, biogeochemistry. Biogeochemistry, if you're wondering what that is, it's the intersection of the fields of biology, geology, and chemistry. And as I said, everything we do after this is we try to integrate things back towards using phages, let's say, for health or for sustainability. Some applications of this might involve using phage therapy to combat antimicrobial resistance, or maybe even use phage therapy for environmental health. When you think about microbiomes, what is the role of phages inside a microbiome? Now, for the longest time, we ignored viruses and phage in microbiomes. When we thought about microbiomes, the first thing that came to mind was bacteria. And I think bacteria have been studied in the context of microbiomes, but have phage and viruses been studied? The answer in general is no. I would argue that viruses, specifically phage, for the most part have been ignored in microbiomes, in ecosystems, in ecology. But if you think about what a microbiome is, there are viruses, there are bacteria and archaea, there are eukaryotes, and all of these are helping turn the ecosystem around, trying to cycle nutrients, important in the context of biogeochemistry, really, really important for the ecosystem overall. What my group is specifically interested in doing in an environmental context is try and work towards an integrated understanding of biogeochemical cycling in the ecosystem. So the idea is that there is a holistic sort of interaction between biology, geology, and chemistry, where viruses, specifically phage, are playing an important part. So we want to take the microbiology and the biogeochemistry, and then especially right now with, let's say, all of the longitudinal work that we are doing, we want to move towards a predictive understanding of ecosystems. So essentially, we are making observations today so that we can better predict responses to the environment, ecology, evolution, whatever you want, better in the future. My group also develops tools to study microbiomes and viromes. I say tools, I don't say software, because we're also interested in developing algorithms and databases. We're not just developing software, but software is one facet of what we do. Here's just a snippet of everything that we have developed so far. Much of this is in the context of viruses and phage, but we do have some tools that we've developed broadly for microbiomes, such as metabolic here, and then there are some that are being developed. So some of my students are here who would love to talk to you about some of these. So PST is a deep learning model for studying viruses and phage, developed by Cody Martin, a student who has a poster out here. And CheckAMG and Fox are databases and software for viral ecology that are being developed by James Cosmopoulos, another PhD student at the lab. So with this sort of background on our lab, let's think about freshwater. And this is what I'd like to talk to you about today. So let's think about where Earth's water is. So there is water on Earth, but the majority of the water is in marine systems. Oceans constitute about 96.5%. So fresh water, all the fresh water on Earth constitutes about 2.5%. If you break this down, this fresh water, surface fresh water, that is lakes, are about 1.2% of this. The rest of it is locked away in groundwater or in glaciers. Now if we break this down, surface fresh water, much of it is actually frozen. Much of it is frozen. So if you think of it, how much water on Earth is actually present in freshwater lakes, which are incredibly important socially, right? It's only about 0.006%. Only 0.006% of all water on Earth is held in freshwater. Yet it is important socially, it's important for humans to survive. I mean, just imagine the context of that. When you think about freshwater, much of the freshwater is present in a climatic context. So there are 4 broad climate divisions on Earth. So there is tropical, so there's tropical lakes. There are dry climates, so these would be, let's say, deserts. Then there is temperate or continental climate. So much of the US, for example, is present in a temperate sort of an environment. And then finally, there's polar climates on either poles. So if you're studying freshwater, if you're studying lakes, you have to study them in some sort of a context like this. In my lab, we primarily study tropical and temperate lakes. So we have field sites and model systems that we use to study both of these environments. Lake Tanganyika is in Africa, so we access it off of Tanzania. This is one of the deepest lakes in the world. It's a very pristine lake as well. So we study, this is a tropical lake that we study, and on the second one you're seeing is Lake Mendota in Wisconsin. This is the model system that I will be talking to you about today. Now, why should we be studying freshwater lakes? So before I show you any data, it's important to recognize that freshwater is a resource that is rapidly declining in quality. Oxygen in freshwater is declining at an alarming rate. I mean, oxygen worldwide is decreasing. It's decreasing because as temperatures go up, the amount of oxygen that can be held by water decreases. And this is true for the oceans, this is true in freshwater, but in freshwater, it's declining 3 to 9x faster than in the oceans. And the cycle that this sort of drives is temperatures increase with global warming. This leads to increased stratification, so the deeper layers of water lose oxygen much quicker than the upper layers. Once the oxygen is depleted, microbes come about. They can respire other forms of electron acceptors. They can respire nitrate, they can respire sulfate, they can respire carbon dioxide. So they'll produce greenhouse gases like methane or nitrous oxide and toxic gases like hydrogen sulfide. And the combination of all of this manifests in, let's say, something like fish kills. So if you see this is a manifestation of loss of oxygen in the water. And so you increasingly observe this. So essentially pollution is being driven in freshwater lakes due to increased warming as well. Lake Mendota in Wisconsin that we study, if you're thinking about the map of the US, this is Lake Michigan, this is Chicago, and Madison is located right here. Lake Mendota is an urban eutrophic lake, which means that it is an urban lake. It's in a small city. It's eutrophic, meaning it's very rich in nutrients. It's a temperate lake. So as I indicated, it's in the higher latitude. So it's a temperate lake. It's dimictic, which means it mixes twice a year. Some lakes never mix. They're just stagnant. Other lakes mix. That's what this means. And then we have a lot of chemistry data. So we've observed that the lake has low iron and high sulfate. What's very interesting about Lake Mendota is that the University of Wisconsin is home to the oldest limnology department on the planet. So this lake has been continuously monitored since 1889. So it's considered to be amongst the best studied lakes on the planet. And since 1983, it's funded by the US government to maintain a long-term monitoring program. So that's where a lot of our data comes from. Sampling Lake Mendota is an incredible process. A lot of our students have the privilege of being able to sample here. So shown here are 2 of my students. One is a former PhD student, Patricia Tran, who, as you see here, is sampling in over ice. So you have to drill through the ice and then you have to sample the lake. Shown here is a former IIT Madras master student who's currently a PhD student with me, Dinesh Bhaskaran, who is sampling for viruses in Lake Mendota. So we've maintained time series of bacteria primarily, but increasingly we are also maintaining time series of viruses. So as you can see, in addition to sort of learning about microbiology, an integral trait that we look for in our students is bravery, because you have to be able to go on the ice and sample like this. So what I'm going to talk to you about is a 20+ year time series. So this is now year 26 that we are maintaining a time series. We've essentially gone there every 10 to 14 days and sampled. So starting in around 1999, 2000, what we have analyzed so far is about 500 samples. These primarily comprise the water column, so we just essentially put a sampling device down and take a sample that integrates the top 12 meters of the lake. We have a buoy in Lake Mendota that takes continuous measurements of a lot of things like pH, temperature, physicochemical parameters, chlorophyll, and so on. So we can get all of these concentrations in organics, metals, chlorophyll, DOC, temperature, phytoplankton and zooplankton biomass, and so on. Now, when we've conducted 16S RNA amplicon sequencing with our collaborators, typically our collaborators have noticed that there is 6 seasons that we can sort of track the microbiology with, and generally speaking, microbiology follows a very predictable pattern. So to build on these studies, we've conducted metagenomics and then tried to study viruses from these studies. Our typical process for this is very analogous to what we would do for bacteria. We get our sequencing assemblies. This is all metagenomics, so sequencing all of the DNA. We identify the viruses. We identify viral genes. We identify the viruses. We reconstruct viral genomes. We observe the genome quality of all of the viral genomes that we reconstruct. We conduct phylogeny so that we can taxonomically identify the viruses. We predict the hosts of the viruses, we annotate the genomes, and then finally we conduct genomic and ecological analyses to understand the roles of the viruses. So now let's look at this through the few different lenses I sort of mentioned initially. So let's think about viral diversity. Over the course of 20 years, sampling at the exact same spot in the lake every 2 weeks, we recovered about 1.3 million viral genomes. So we also have some species statistics, so we can convert those as well. But from a genomic perspective, we have about 1.3 million viral genomes. So about 2,600 viruses per sample is what we average, and up to 8.1 proviruses. So that's prophage, 8 prophage per sample. One interesting thing I will mention is that when we have constructed, or say computationally constructed viruses, typically the idea is typically what's been done in the field is viral genomes have not been reconstructed accurately through a process known as binning. In fact, I'm here to encourage the process of binning for viral genomes, because if you do bin, that is you reconstruct your viral genomes and you curate them, what you observe is that you will get sequences that are 4 times longer in length and have 3 times higher genome completeness. So if you curate your genomes and your genomes are longer, you can recover more viral proteins and your ecological observations will be much better. So this is something that's not traditionally been done for viruses, but we do think it should be done going forward. What is fascinating is that even after 20 years of sampling the exact same spot in a lake over all of the seasons, the viral species rarefaction curve did not plateau. So what this is saying is that there is vast diversity even in that single lake that remains yet to be discovered. So even after 20 years, so let that just sink in for a second as to how much viral diversity can exist in just one lake. When we look at the viral communities, what we observe is that they differ across seasons. So we can divide the entire year into 6 different seasons, which we call ice on, spring, clear water, early summer, late summer, and fall. And what we observe is that when we look at the viruses, the majority of the viruses belong to double-stranded DNA viruses from the family known as Cordyceps, but we can recover eukaryotic viruses as well. And when we observe these viral communities, they cluster very cleanly by season. So for example, if we just focused in on this one plot, there are 3 colors indicating viral communities that are sourced from 3 different seasons. And as long as you see separation between the data on this particular 2D plot, that's an indication that the communities are very different. So essentially the communities from the fall here are very different from the spring, and you can extrapolate that to other samples as well. Now thinking about viral ecology, my lab has been very interested in a specific process known as auxiliary metabolism. And what auxiliary metabolism is a way to manipulate the hosts of viruses. So how do phages impact their hosts or impact nutrient cycling? So think about it very simply. A virus lands on a bacterial cell, and as soon as it lands on the cell, it injects its DNA, it begins the infection process. And once the infection process is sort of finished, it's going to manipulate its host, and once this host dies, all of the cellular contents of the host are sort of available to the community. Now the rest of the community can take these ingredients, let's say phosphorus, sulfur, carbon, nitrogen, And that is a way they can use these ingredients, and that's the way the community is impacted. But beyond this simple infection cycle, what phages can also do is carry what is known as auxiliary metabolic genes. These genes are acquired from their hosts, they are retained, they are functional, and they offer fitness benefits. What phages typically do is exploit these AMGs to exploit specific niches. For example, phages might exploit the niche of photosynthesis, might exploit the niche of methane oxidation, very specific processes. These AMGs, or metabolic genes that are present in phage genomes, are not involved in essential phage processes like replication or lysis. So from our 1.3 million genomes, we recovered about 140,000 auxiliary metabolic genes. When we sort of clustered these, we found that there are 575 protein families, including families that had never been found before. So this is quite interesting in a way. What we observed is that viral populations were quite heterogeneous. That is, in a viral species, there were some strains that carried AMGs, and then there were others that did not carry them. And the distribution of these varied by seasons. So in some seasons, you would see that viral species, or the most abundant groups of viruses that infected a particular host, carried AMGs, and in others, they did not. Since AMGs can confer fitness benefits, we looked at their distribution in the context of seasons, and we saw that their distribution was strongly associated with seasonality. And what you're looking at over here is just a snippet of all of the different processes that these AMGs are involved in. But just to give you a sense for it, there's photosynthesis, there is methane and a lot of different associated metabolism, there is sulfur metabolism, carbon fixation processes, and finally nitrogen metabolism. Now, what can we learn from this? Sure, there's a lot of diversity. I showed you viral diversity, I showed you protein diversity here, but what can we actually learn from this? So let's look at this from a few different contexts. The first realization we had was that when you observe the 575 protein families we had, they're not all metabolic. So this is work of one of my students, Cody Martin, which shows that when we think about metabolic genes, I think it's a misnomer to only call them metabolic genes. In fact, they're auxiliary viral genes. And when you think about these, the auxiliary metabolic genes are just a fraction of these. The other kinds of genes that we typically see are genes that are associated with regulation and genes that are associated with physiology. So these genes, these auxiliary genes, are manipulating their host in much more sort of diverse ways than simply metabolism. So essentially what Cody has developed is an eco-evolutionary framework where we can look at different eco-evolutionary forces that seem to be driving the evolution of auxiliary viral genes. Another context that we can look at is we can track the distribution of viruses over both time and season. So over a single year, we can see how viruses change over every month, every season, as well as we can observe how they're sort of stagnant, how they're persistent over 20 years. So for example, we can look at different viruses that infect a single host. So here's a snippet of an example. Your red, so this plot over here has 3 colors. There are 2 different bacteria I'm showing. Both of them are cyanobacteria. The red plot indicates hosts, and the graphs in green and blue are viral. So these are all abundance plots. So the y-axis is showing abundance, and the x-axis just goes from Jan to December. So just showing you average of the 20 years. So you can see that in this case, they're quite detached. The host sort of has a certain kind of abundance, the virus has a very different kind of abundance. Over here, they seem to be sort of tracking each other. So there's some sort of synchronicity to the distribution of the host and the viruses. And this is a pattern I'll get back to again. Now, what's interesting over here is that this sort of pattern we can develop for every single one of the hosts and every single one of the viruses. And for example, if you're tracking a single cyanobacterium, we've identified up to 350 viruses that we can track over time. So you can do this for viruses, you can do this for bacteria. If you observe the abundance in the distribution, what we find is that there's 2 different kinds of patterns. There's viruses that we call persistent viruses. They're present across the entire 20-year data set. They're present in every season, every month, they're always there. The abundance changes, but they're always there. The second kind of viruses are what we call temporal viruses. These are very seasonal. They come up in a season and then they sort of disappear. They are just not very abundant, let's say in the winter. They're very abundant in the fall. That's what you're seeing. These viruses infect specific kinds of organisms. For example, these would infect methanotrophs. if you now begin to look at these dynamics, what we observe is there's two different kinds, and we can look at both the DNA and the RNA. So if you look at the DNA, you observe this, where there is a host and there's a virus. Let's say the host is shown in dark lines and the virus being shown in sort of the lighter lines. You observe that over here, both of the patterns are synchronized with each other. Whereas over here, you'd see patterns where the host and the virus abundances do not track each other. So this is something we're referring to as asynchronous or desynchronous. You can observe this in both DNA and RNA. So what can we do with this data? We figured out, we think what's happening here is that the synchronous data refers to what is known as the piggyback the winner model for viral genomics, viral ecology. What this suggests is that there is a certain kind of virus that integrates into the host genome in the form of a prophage. So these are temperate phages, and these develop these sorts of patterns. So, on the other side, there's a model known as kill the winner, where the most abundant viruses typically infect the most abundant hosts, and that is manifested in the form of these kinds of observations. But now this beckons a question. Are these the only observations we see? The answer is no. This only explains a small subset of our observations. So while we have used these sort of observations to fit our data into these two existing models, we're also testing and developing new models. We are building off of the existing models, but we're also testing and developing new models. So with this, I'll just summarize everything I showed you. We did discover a lot of diversity over the course of 20 years of studying a single freshwater lake. We have found lots of positive associations between AMG-containing viruses, and this is in the context of seasonal and temporal change at multiple levels. I have not had the opportunity to talk to you about anything to do with evolution, but we see that there is selection for viral genes associated with fitness. We can observe soft genome-wide sweeps through the populations, through different viral populations, in fact. We also have tracked the association of different inorganic compounds with different viruses, as well as through virus-host relationships. And these papers, both the virus paper as well as the companion paper, which was done in collaboration with Brett Baker, who's here in the audience today and you'll hear from today, are available on Nature Micro. So please go ahead. The papers and all of the data sets, all of this 20-year data set is public. So I encourage you to use these data sets and ask interesting ecological or evolutionary questions from them. I will acknowledge funding and collaborators, Trina McMahon and Brett Baker. All of the sequencing here was provided by JGI. So I'm thankful to JGI, NSF, and NIH specifically for funding this. And grateful to all of the PhD students and postdocs from my laboratory who have assisted with this. Everything you saw over here is analysis that was conducted by Chao Zhu, who was a postdoc in my lab and is currently an associate professor at Shenzhen University. And Patricia Tran, who was a PhD student with me and a research scientist at UW-Madison, and Sam Bashand, who was my lab manager and is now a PhD student at Stanford University. And with that, I'll thank you, and I'll leave this out. These are posters from my students, so I'll take any questions.
Thank you very much, Karthik. And it is tough to put all 20 years worth of work in 30 minutes. And I'm going to request two from audience, one from the online people. Any question here? Yeah.
Hi Karthik, yeah, great talk. So just a quick word about the origin of this lake. So it's a natural lake, like, or the water is coming from somewhere? Like
the lake is natural.
Natural, so just the rainwater.
Yeah. So it's rain, it's primarily precipitation, snow melt, and
Okay. So obviously there is no contaminant as such, like which impact the seasonal pattern of any viruses or bacteria.
So we can track it. So for example, we know that there have been multiple. So there have been infestations in the lake. So for example, zebra mussels or certain insects can get in, and those years we see that the microbial community gets affected, and we can detect those. So when we know that there is, you can see that in the data.
Right. No, and especially I'm not sure. Like you discussed like till 2016 or 2020. Also, you discussed some data. Any pattern which you find unusual during COVID, as such?
No, that's a good question. So So we weren't looking specifically for coronaviruses. That's not something we've done, at least not yet. But we have patterns that we've observed in, let's say, in 2014, there was a spiny water flea, which is a specific kind of insect. There was an infestation that impacted the communities. I'm sure if we went looking, we would see something with COVID.
Yeah. You have viral DNA stored, so you can go back.
We have it stored, yes.
Please.
Thanks, Karthik. I was wondering whether you ever saw or how many virus genomes that went kind of extinct during the time course and how many new ones arose because your rarefaction curve is rapidly ascending.
That is a great question. Do we see extinction and reappearance? The issue is we can never be sure. When something, the problem we have is with detection. So if something appears all of a sudden, where did it come from? Was it a contaminant? Did it come from a certain source? Or was it present but abundant at very low levels and then suddenly became abundant? That's the only reason I don't talk about extinction. It's just very difficult to be confident that we are saying the right thing.
Okay, so last question from this audience here. Go ahead.
So it's a two-part question. Sorry, Karthik. I am very new to the viral ecology. I've dealt with microbes and 16S work. So my first question is that the viral diversity, like how much viral mutations actually influence the viral diversity and how is that incorporated in the entire bioinformatics pipeline? Is it ever incorporated in the first place? The second one is, of course, contamination. So I'm sure a lot of Indian scientists would like to repeat this work in Indian lakes, in Indian rivers, and how do you foresee this work sort of getting translated into contaminated Indian waters?
So I'll answer the second part of your question first, which is that this work can stand on its own in any system. This is a method that we've applied in a certain system, and I would argue that today work by our lab as well as many others has developed really solid methods for viruses and phage. So if you wanted to take those methods and study anything, let's say the human microbiome, viruses in the human gut, viruses, let's say in a contaminated lake, even a radioactive lake, you can do it. As long as there is DNA there, as long as there's RNA there, you can study it. You just have to extract the DNA, sequence it, you can study it. So there's no worries or there's no barriers to adopting it in a contaminated lake here. There's no barriers whatsoever. The first part of your question was mutations. With mutations, what I will say is that we capture every single mutation that's present. So when we assemble this data, both in sometimes in the assemblies, many times in the reads, but through a combination of these, we capture every single mutation that exists. So when you're looking at the viral genomes, we can give you all sorts of SNPs that are present. The question is, how do you make sense of it? That's a different question altogether. But as far as capturing the mutations, we can capture it. Can we make sense of it? That's a different question altogether.
There are many more questions maybe there. Let's take the last one from the first, how does one map the phage accurately? Go ahead.
How does one map the phage accurately to the bacterial host? Do we observe co-evolution of, that's a great question. The answer is, how can we map the phage to the host? There's a lot of methods. The best method I can sort of describe is CRISPR-Cas systems. CRISPR-Cas systems in bacteria and archaea encode a small snippet of the phage genome as a memory against past infections. So you can use those to sort of link individual phage to individual bacterial or archaeal hosts. But you can also use other methods. There is different methods. So for example, the AMGs I showed, that's an interesting method because that's very host specific. There's schema-based methods. There is lots of methods, lots of tools available to sort of automate these processes as well. And do we observe co-evolution of host and phage? The answer is again, yes. We observe it in multiple contexts. When we are talking about AMGs, there is an arms race between bacteria and phage. Phage also steal a lot of genes from their bacteria. Phage are also sort of fighting back against bacteria. So we can observe co-evolution in multiple contexts. I'm happy to talk about that more in person later.
Thank you, Karthik. A big round of applause for him.
Hello. Our next speaker is Dr. Vatsan Raman.
Okay, welcome back to session 2, Ecology and Evolution in Microbiomes. This session will be chaired by Dr. Gaurav Sharma and Dr. Aarti Ravindran. So to quickly introduce them, Dr. Gaurav Sharma is an assistant professor in the Department of Biotechnology at the Indian Institute of Technology, Hyderabad. He completed his PhD in microbial genomics from the CSIR Institute Microbial Technology, Chandigarh. His postdoc work at the University of California, Davis, centered on the evolutionary origins of various microbial systems such as type 4 pili and motility mechanisms in myxobacteria. He's also been recognized for his contributions to science, winning awards like the AMI Young Scientist Award and the BioClose Innovation Award. Dr. Aarti Ravindran obtained her MSc Biological Sciences and ME Biotech from Birla Institute of Technology, Pilani, Rajasthan. She also earned her PhD from Anna University, where she worked on systems biology, phylogenetics, comparative genomics, and molecular dynamics simulations. She is currently working as a postdoc researcher with Professor Karthik Raman here on metagenomics, machine learning, and metabolic methods of analyzing environmental and gut microbiomes. Over to the session, Jess.
Okay, so welcome to Ecology and Evolution in Microbiome session. So we have 3 speakers this time, and all stalwarts in their field. So let me introduce first speaker, Brett Baker. So Brett Baker is an associate professor at University of Texas, Austin, and he holds positions in both the Department of Marine Science and Department of Integrative Biology. He is a recipient of Dwight W. and Blanke F.A. Reader Continual Fellowship in Systematics and Evolutionary Biology. He earned his PhD in Earth and Environmental Science from University of Michigan, and his research focuses on microbial diversity, ecology, and evolution, particularly in marine environment. So throughout his career, Dr. Baker has contributed extensively to the scientific community, and we would be very happy to listen to what he will be presenting today. So yeah, over to you.
Thanks. Hello? No mic? Hello? I can yell. Oh, there we go. Okay, cool. Yeah, so I'm going to switch gears quite a bit from what we saw this morning. Going to go both to Archaea and, no, I don't want subtitles. Go away. There we go. And talk about their role in our understanding of the origin of eukaryotes, or the origin of complex cellular life. I would like to start by this quote, which one of my sort of early formative years really enjoyed Carl Woese's writing, and so I found this very accurate, considering it was written in 2000, where he says, "The deepest branching of the tree of life takes us into uncharted evolutionary waters. The door to understanding early, more primitive lives has opened." Right, and so this all sort of started with a work by Anja Spang and Thijs Edma. At the time we're at Uppsala in Sweden, where they were reconstructing genomes from Loki's castle hydrothermal vent in the North Atlantic. And they came across this organism that they named after the vent called Loki Archaea. And so this sort of is placed in the tree of life. It's sort of like, at the time, was considered sort of our missing gap between microbial life and eukaryotic life, right? So this was a game changer, right? So at the time, I was doing similar work using metagenomics to reconstruct genomes from other places, and went running into my grad student's office and said, "Oh my God, we have to check this more," and we had something very similar, and that was another organism that we ended up calling Thor Archaeota. So continuing the Norse mythology, or MCU, whichever you prefer. And we came up, Thijs and I teamed up now 11 years ago to, instead of trying to compete with each other, we have had a very good collaboration for many years to try and reconstruct more of these genomes. So at the time when Loki came out, there was only one genome, and at the time this paper came out in 2016 or 17, we had 9 or so, and so we named them under, at the time, phyla. So Odin, Thor, Loki, and Heimdall. I'll point out that Heimdall, based on this tree, appeared to be the closest sister group to eukaryotes, right? And so Jill said that we are Borgs. Well, I'm going to say we are Asgards. We may be both, but it's my perspective. Why was this interesting? Why was this exciting? Why was this so controversial? Because if you open up a biology textbook, you see something like this, right? Prokaryote, eukaryote, very different in terms of cellular structures, right? The trees themselves, I was skeptical, as most people were. But what was really compelling was the fact that they had a suite of genes and proteins that had only been seen, many of them had only been seen in eukaryotes before. Things involved in cytoskeleton, protein trafficking, and so on, right? So if here, this is essentially showing on the top the 4 file that we had at the time, and eukaryotes, and then other archaea. So you can see some of these are randomly distributed within other archaea, but there's a huge distribution of more of these eukaryotic proteins within the Asgards. Based entirely on genome annotations and functional predictions, this is sort of an overview at the time of what these functions were, right? So a lot of this sort of like, hallmarks of a eukaryotic cell, these archaea were having, right? And I'll particularly note the escort proteins, and I'll come back to that in a second, why they're really important. At the time, this was just genome prediction. In recent years, there's been a whole bunch of papers that have taken these genes, expressed them in different organisms, and shown that they function very similar to the way they do in eukaryotes, in this case, profilin, which regulates actin. This great work by Bob Robinson. This idea of us evolving from archaea was not, it's been around for a while. And in this case, this is the first paper that I could find, this was entirely based on very little information, essentially looking at membranes and to some extent physiology. And there were other people like James Lake who also thought in a similar vein. But what does this mean, right? At its core, it's essentially you're looking at, we had really good information about the mitochondria, which evolved from some sort of alpha proteobacteria-like bacterium. We didn't have much information about who the host was in that interaction that led to the first eukaryotic cell, first mitochondria-containing cell. So now we have this sort of Asgard as something that we can look into, try to understand what that interaction was, right? That is what we are trying to get at now, in trying to understand what interactions between this alpha proteobacteria and the Asgard led to the first eukaryotic cell. Early on, we used genomic prediction, and it appeared to us that, again by Anya Spang, that there was some sort of interaction perhaps within electrons, hydrogen, acetate, or formate, right? And then soon after that, a Japanese group isolated a Loki, and sure enough, it has an exchange, as far as I can tell, based on genome prediction, and it was a co-culture with a methanogen and a sulfate reducer. What's really interesting is when they sort of looked at these cells initially, they were just spherical, didn't seem to have much structure. As they did better electron microscopy and looked at over different stages of its life, it sort of started to form these arms, which we now know are full of actin filaments. Very interesting, and we think that may have a role in the interaction with the proteobacteria. So we sort of expanded things. We were trying to get more genomes to understand which group is most closely related with eukaryotes, and to understand the sort of evolutionary history within this group, this larger group, right? So this is from now 2 years ago almost, where an expanded genome database where we showed that this group that we called HOD, we didn't call it HOD, someone else named it, appears to be the most closely related to eukaryotes. And we've refined this phylogeny, mostly Tice's lab, has refined this phylogeny to show that this is really, seems to be the only scenario. We do not really see any other group relating to eukaryotes and being related to eukaryotes in the tree. This was, I will point out, this was originally Heimdall, so the larger group Heimdall. So from the beginning, like these phylogenies have sort of pointed to the fact that HOD is the closest group. What's really interesting is if you look at HOD compared to the other groups, they tend to have more of these eukaryotic proteins. So it's telling us something. We've also done work doing ancestor reconstructions, essentially looking at the gene content of the different groups. And if you sort of think of the Asgard ancestor sort of moving back in the tree, closer to eukaryotes, you go from sort of a chemolithoautotroph organism to more of a heterotroph. So more of like a eukaryotic type metabolism was our idea. We've continued to collect more genomes, as I do, And so the hotbed of where we find a lot of Asgard genomes is Guaymas Basin, which is in the Gulf of California. It's a really interesting place, very dynamic. You can go from microbial mats that are 100 degrees Celsius to a few centimeters down to just plain sort of bare sediments. And this is sort of showing heat maps of that, right? So for whatever reason, this place is really good for getting Asgards. And so we've done a lot of sequencing here. We're now doing a lot of sequencing. Oh, so essentially, what I'm going to talk about now is sort of all of our unpublished stuff that we're publishing soon, but we've expanded the database pretty significantly now, where there's around 450 publicly available genomes. Now we have about 1,000, over 1,000. My lab gave it a cute name, Asgardian, but whatever. What's interesting is they're everywhere, right? So they're not just in hydrothermal vents. You see these things in lakes. You can go to your backyard. Jill's backyard, we found some. They're everywhere, right? And so, as Jill mentioned, closing genomes is good. So there's been some work by Tice's lab, the Japanese group, and Jill, in collaboration with Jill, Luis and Jill's lab, they closed some more genomes as well. We also have viruses that are associated with many of these. So what does the tree look like now? It's expanded, of course. It's there's a lot on this tree that I won't go through, but what I sort of want to show you is that we've essentially doubled the amount of diversity in the last few years from Guaymas, and mainly from Guaymas, to find more of these Asgards. So here now is HOD, this larger group. Around the eukaryotes is up on the upper right there. I guess I can point. Eukaryotes here, here's HOD. So these groups here, Kari and GERD, appear to be the closest relatives to eukaryotes, okay? I want to put this in context, right? And so at the time that the first eukaryotic fossils were seen was soon after the Great Oxidation Event, where there's, sort of a misnomer that it was sort of a gradual oxidation event, but essentially oxygen, we think, had to be in play, so it's a high energy metabolism that we now, sort of think that in general terms that gave rise to larger organisms, right? So we think that having the Asgards, the first eukaryotic cells around 2 billion years ago, it was right after the Great Oxidation Event. And so we've looked at HOD and Kari, those groups that are most closely related to eukaryotes, to see what their metabolisms are, how they compare. And interestingly, they appear to at least be able to tolerate oxygen, but we think they can actually use oxygen. So this is Loki. It was renamed to Promethearchaeum. Up here, fairly simple in terms of its electron transport chain. If you look at HOD and Kari, they have full electron transport chain, complex 1, 2, 3, 4. They have raw, so they can deal with oxygen stress. Pretty interesting, because the models of eukaryogenesis and us, our mitochondria are using oxygen. So why would the host need to use oxygen, right? So this is not really in sort of this didn't account in any of the models for eukaryogenesis that are around right now, essentially. So it's a big surprise. So we looked at the phylogeny of these COX genes and other aerobic pathways, and essentially, so here's alpha proteobacteria here's all the Asgards. It's not simple. And I can't really say that they're ancestral to mitochondria. We don't know if they sort of provided these metabolisms to alpha proteobacteria, but it's interesting nonetheless. So we're sort of updating the model based on these new findings. There's a lot here, and I won't go through all of it, but essentially what we think is that the earlier Asgards lived in an aerobic environment, and then they sort of moved after the Great Oxidation Event to be able to use oxygen to some extent, or at least tolerate oxygen. And that is where the aerobic alpha proteobacteria live, so they partnered up at that point. We think that the increased surface area of the cell, we think that the archaeal host, in essence, had some basic aerobic metabolism, and then the alpha proteobacteria sort of enhanced that. That's our sort of working model. So I'm going to put this back in perspective, right? So we're looking back 2 million years of evolution, right? Unfortunately, we don't have a time machine. We can't get DNA from that long ago. But right, so here, the closest things we have to sort of look at things are lipids, right? So DNA only goes back so far. Origin of life is around 1.5 billion years ago in the Proterozoic. And there's isoprenoids that have been shown by Jochen Brox in the rock record. So we're interested in sort of trying to, see if we can find biomarkers to find Asgards in the rock record, and also understand if Asgards were the archaeal host, did they confer some of that membranes to the host as well? So what is the origin of eukaryotic membranes, essentially? This is a great collaboration with Paula Wieslander at Stanford. This is sort of an overview, right? So lipids are a lot of things. They're involved in signaling, transport, membranes, storage, pathogen interactions, which I'll get to. So we set out to sort of, so first thing we did is to see, sterols are sort of they're a eukaryotic feature, right? Some bacteria form sterols. And there's actually some compounds in the rock record that people haven't been able to identify. So we went through all of our Asgard genomes to see if perhaps if Asgards can produce sterols or hopanoids or some sort of precursor to sterols, essentially. We got very excited because when you look, this is essentially the pathway for sterol biosynthesis. Here's Asgards, here's other archaea. We saw that there's a lot of the steps for sterol synthesis were there. It's like, oh my God, they can make sterols. This is amazing. But when we look, and even when we looked at the alpha fold structures, they're very similar to eukaryotic sterol biosynthesis. So Paula Wielander's lab expressed these genes in E. coli and some methanogens, and in fact, they don't actually make sterols, they make diterpenoids. And if you look out on a tree, they sort of fall, they're ancestral to plant diterpenoids, which is super interesting because a diterpenoid is a cyclic compound that archaea haven't been shown to make any sort of cyclic lipids, essentially. So here's essentially the output in vitro analysis of the products of those enzymes from HOD, actually. And it's interesting because the only other prokaryote, bacteria or archaea, that make a similar compound are mycobacterium. And so here's essentially the compound. It's very similar to this thing that mycobacterium makes. What does this mean? We don't even know if this lipid is in the membrane. It could be used for something else. So we have been looking at what is the role of this compound in mycobacterium. And it turns out it actually sort of mediates infection within its host. It sort of stabilizes the endosome. So when cells are in the endosome, it stabilizes that environment. So we think, and I'm going out on a limb, but I think that perhaps this lipid may be involved in somehow interactions with some host that we don't know what that host is. We're now working on trying to understand who the host of HOD is. There's no cultures that are published. And so it's super interesting. So there's people in my lab here. A lot of this work is Katie and Val's. Collaborate with Jill, a lot with Tice Etma, and Chris Greening and Paula. Thanks.
Okay, thank you very much, Brett. Any question? Yeah.
Thanks, Brett. That was great. I was wondering, do we not need to look more than at intracellular pathogens to understand, mechanisms underlying endosymbiosis?
Yeah. It's sort of a new revelation to me that they have this compound, and I need to do more reading on what's known about this compound in mycobacterium. I don't know. Yeah, yes, absolutely. My answer is yes.
Because I had never seen it framed as intracellular pathogenesis, right?
Yeah.
Oh, all right, thanks.
Yep.
Any other question?
So, sir, of all the archaeal genomes that you are showing in your study, like 1,000 archaeal genomes that you have, so are these all metagenome assembled genomes?
Yes. Well, yeah. Not all of them. There's 2 isolates of Loki, but the rest are metagenome assembled. Yep.
Okay, so were there any specialized methods for binning these archaeal metagenome assembled genomes, or it is similar to the bacterial?
Yeah. So yeah. I mean, we've used multiple binning approaches, and then used DOS tool to do that, and then we do downstream curation, and there's a lot of people that spit out a lot of genomes, and these genomes we need to be very careful with and make sure they're not contaminated, because if there is any sort of contamination, it could be the story behind Asgard, which would be harmful to our whole situation. We curate our genomes. Yeah.
Okay.
Does that answer your question?
Yes.
Jill's got a question after that one. Yeah. Yep.
Yeah, so I did some of those genomes and completed them, and long reads were really important.
Yep. Yeah, Jill's the one that closed the 2. And,
What are the challenges to cultivate this Asgard archaea?
So Loki's an anaerobe, and it grows really slow in cultures. That's all I can tell you. Hod's a heterotroph and an aerobe, from what we can tell. Should be much easier. Yeah, I mean, many of the places like marine sediments, not Guaymas, but they're very slow-growing environments, right? So, it's the doubling time of the first Loki culture was, I don't know, months or what weeks it was, I guess. So it took them it took them 10 years before they actually realized what they had, and they can't. All those Loki cultures are slow enough that they can't actually do any physiology on them, right? Yeah, I'm collaborating with people to change that. Stay tuned. Yeah.
So my question is probably similar to his.
Yeah.
Does the genome, can the genome inform like metabolic, as in culturing techniques which can be employed to isolate them, or at least bring them in culture, co-culture?
Absolutely. Yeah. I mean, this was. I mean, Gene Tyson and Jill's lab, was the first one to take a metagenome, yes, and show that it was something. It was a nitrogen fixer from acid mine drainage. We're doing a similar thing with HOD. Okay. Yep. Absolutely.
There is no other question. Thank you very much, Brad.
Thanks.
Yeah.
Good afternoon. It gives me great pleasure to introduce the next speaker. Dr. Shilpi Sharma is a distinguished professor at the Department of Biochemical Engineering and Biotechnology at the Indian Institute of Technology, Delhi. She earned her PhD from the Ludwig Maximilian University of Munich, Germany in 2004, focusing on plant microbe interactions. In 2008, Dr. Sharma joined IIT Delhi, where she has been instrumental in advancing research in plant microbiome interactions, Rizosphere engineering, the development of next-generation bioformulations. Her notable contributions have been recognized with several accolades, including being named among the top 75 Indian women in STEAM, science, technology, engineering, applied arts, and mathematics in 2022. In 2023, she was honored with the Tata Transformation Prize in Food Security for her innovative research on microbial diversity and its applications in environmental sustainability. So welcome you, ma'am, to give a talk.
So, thank you, Chair. I hope I'm audible to the backbenchers as well. Okay. Thank you, Karthik Square, for the invitation, especially to talk in such an august gathering focused on microbiomes in different habitats. I'm again going to switch gears. I've titled my talk as "It Takes 4 to Tango." I would have probably a couple of years back titled it as "It Takes 3 to Tango." Right now I'm going to talk about harnessing the nexus between plant, its associated microbiome, soil, and the environment, soil being now considered as a separate vertical. And I'm going to talk about how this can be harnessed for agricultural sustainability. The overall goal of the group is that we're trying to address some of the challenges that are being faced by agriculture worldwide. Of course, there are increasing number of mouths to feed, but the limitation here is that the arable land is deteriorating. And I have to admit that India does not have a really good number in terms of the per capita arable land available. So we are working towards using microbiome-based solutions for food security and sustainable agriculture. Now, when you talk about agricultural sustainability, of course, food security happens to be prime, but we have to look into this in the context of several terms here. We're looking at environmental sustainability, we're looking at biodiversity, we're looking at plant health, but we're also looking at soil health. Now, we have been talking about, there have been some talks on the gut microbiome. Similar to that, you know that every higher organism is now looked at as a holobiont. In the case of plants, you will have the good and the bad. The good here is the one responsible for the fitness of the plant, its overall productivity when you're looking at agricultural crops. But then there's also a set of the bad microbiome, now termed as the pathobiome, which leads to the manifestation of a disease. And there is a strikingly similar analogy between the gut microbiome and the special zone of soil where I'm going to focus on, called as the Rizosphere. Now, the Rizosphere, as the name suggests, is the zone surrounding the plant roots and is directly influenced by the roots. The analogy is not just in terms of the habitat that the two are characterized by. So, for example, the gut and the Rizosphere are open systems with large surface areas, and you will have them overpopulated with microbes. The gut and the Rizosphere are characterized by a gradient of oxygen, water, and pH, resulting in the diversity of niches. And the gut and the Rizosphere microbiome structures are shaped according to the host genotype and the age, these two being just some of the few factors that actually are responsible for recruitment of the microbiomes. They also help in the health and the development and growth of the individual. So, for example, protection against pathogens, production of various enzymes and antibiotics, immune system modulation, so whether it is against the pathogen or even in general in terms of acquiring nutrients, in terms of affecting the physiology, the two have similar kinds of roles. Now, I will be focusing on, like I said, the zone called as Rizosphere, and here, unlike in the gut, you will have a larger component of horizontal transmission or recruitment of microorganisms, and a minor fraction is actually the vertical transmission. So, for example, here, the major bank happens to be the soil. It is the root exudates which actually recruits these microorganisms or attracts them to this zone, and then a subset of this is actually found in the endosphere, which is inside the plant parts. This was in terms of the recruitment. In terms of the functionality, like I said, basically it's responsible for nutrient acquisition. You will see that a lot of stress tolerance is also conferred by this plant microbiome, and there is pathogen suppression. Now, what are the factors which shape this interaction? Well, everything from related to the plant to the soil and everything is in between actually determines both the structure and the function of the microbiome. And there's a thumb rule which actually exists when you talk about microbiome, whether it is the gut microbiome or the Rizosphere microbiome. Dysbiosis leads to compromised health. So there is a bad soil and a good soil, and you can have several plant health indicators and soil indicators, which boils down to the dysbiotic microbiome leading to compromised nutrient turnover and thereby compromised plant health. And a healthy microbiome would have a larger diversity responsible for nutrient turnover, for pathogen suppression, etc. So if that is the case, and there is such a strong correlation between the good microbiome and the plant health, our idea is to engineer this zone to basically transform a non-optimized meta-organism into an optimized meta-organism, which will show enhanced NNP availability, it will show enhanced disease suppression, mitigation of different biotic and abiotic stresses. And this has been attained actually by 3 strategies. People use plant-based engineering of the rhizosphere, wherein they generate transgenics. They could also do cultivar selections for actually playing with or modulating the rhizoexudation, which in turn selects the beneficial microbiome. That is the plant-based approach. Then there is a meta-organism-based approach, wherein there is co-engineering or crop rotation done. You basically do not delineate between the plant and the microbiome. Now, both these approaches have their own sets of challenges. We work with the microbiome-based approach, wherein there are people who consider the application of microbial inoculants also under this approach. This is the more conventional approach, but there's also an approach wherein you work with the microbiome per se. So my talk is going to be focused on the microbiome-based approach. It has been decades that we have been applying beneficial microbes to plant for enhancing their productivity. And these are just some of the organisms which you all might be aware of that are used for plant enhancement of productivity, mitigation of stresses. But there's several challenges associated with applying these individual bioinoculants. For example, there's always, this is kind of a common thing that you'll come across, that there is inconsistency in performance in fields when you take them from the controlled conditions. The shelf life is a question. We are still unraveling the larger picture of mechanism of action of these bioinoculants. The action is slow, the economics is compromised, especially when you compare them to chemical fertilizers. So a lot of my work actually went, especially during the formative years of my working group, we were working towards untangling this nexus, the plant, microbe, and soil nexus, trying to understand what goes into the recruitment of microorganisms, what are the different factors which affect this. And in the last few years, things have changed. There has been a paradigm shift in that, in this typical disease triangle as well. So if you look at the host environment and pathogen, which is responsible for conferring or manifestation of a disease, this triangle has now changed into the disease tetrahedron, which you will see has the host microbiome as one of the vertex. So this is how things have evolved in the couple of years. I primarily work with these 4 crops, mung bean, pigeon pea, tomato, and wheat. There are some other crops as well, but primarily these are the 4 crops that I work with. And the approach that we adopt is both the reductionist approach and the holistic approach. Now, when I say reductionist approach, we follow the conventional approach of actually enumerating the good bacteria, characterizing them for various features, and then trying to bring them together. So either forming biostats or forming consortia, and then applying it onto the plant, which is the rather conventional approach. But we also follow the holistic approach, wherein the idea is to use the microbiomes in toto without really disentangling the interactions or trying to enumerate them. We try to see if microbiomes can be applied to in agriculture. And the other backup plan actually is to generate synthetic microbial communities and then apply them to the plants. So I'll show you some examples of both these approaches that we follow. Of course, this is the bottom-up approach, while the microbiome-based approach is more top-down, wherein we first try to identify the good microbiomes and then apply them to agriculture. I've just listed down some of the projects that we're currently working on, wherein we're using the plant microbiome for agricultural sustainability. The first one is the story that I'm going to talk about in the next couple of minutes, and that is about acclimatization of Rizosphere microbiome for stress mitigation. But other than that, we work on development of next generation bioformulation. So basically, this is to enhance the survival and the efficiency of conventional bioinoculants. Then we also work on a parallel to fecal microbiota transplant, wherein we try to do rhizomicrobiome transplantation as a farmer-friendly approach for agricultural sustainability. And we work towards mitigation of biotic stresses by unraveling the pathobiome. So I will be talking, like I mentioned, about the first story. I'll briefly touch upon the other 3 stories to take you through. Talking about enhancing what is already there in the market by adopting certain strategies. We work towards using metabolite-based formulations to enhance its survival once it is applied into the field. We also work with optimizing conventional additives, and we look at the choice of alternate carriers and bioformulation type. Just showing you one example wherein what we did was we used the conventional cell-based Bradyrhizobium species, which is used for nodulation. And in the endogennetic plane, there's a catch-22 situation wherein even after applying Bradyrhizobium species, there is no nodulation. And because there is no nodulation, the farmers would apply N fertilizers, again completing the loop. So then they would always need to apply the fertilizers. So what we did was we used this conventional strain of Bradyrhizobium and made some formulations with EPS. We used several metabolites. I'm showing you some data on EPS-based formulations. And we also used the supernatant to go cell-free. And just to show you a representative of the kind of experiments that we performed, this is in the experimental fields in IRI, an ICR institute. This was the cocktail that we applied compared to the control and the mono-inoculation, which is just the Bradyrhizobium species. And in fact, this species was also controlling a fungal pathogen, Fusarium. So if you look at this being just the pathogen infestation, and this being the cocktail which we used of the pathogen, the cell, the Bradyrhizobial cell, the supernatant, and the EPS. So this was one story when we were trying to actually enhance the potential of an already known inoculant. Coming to the rhizomicrobiome transplantation, which was again a microbiome-based approach, and this also got some funding from, generous funding from the Tata Sons. We tried to identify good soil. This is a simple strategy. We characterize the microbiome, then we go for a strategic transplantation into the field and see if we are able to transform the bad soil to the good soil. What we are right now working on is actually mapping the nation, the arable land actually, for a phenomenon known as disease suppressiveness. This is the inherent ability of a soil to protect the plant from a pathogen. So even if the pathogen is present, it would not manifest as a disease. We are trying to map some of these states that I've pointed out. We've already sampled those. And we're trying to see if this can then be used to transform the conducive soil, the disease-conducive soil, to suppressive soil. And finally, the short story here about mitigation of biotic stresses by unraveling the pathobiome. Now, the concept of pathobiome is rather new. What we're trying to do in this case is, this is a study with Kajanis Kajan, and we're working at mitigation of a wilt caused by Fusarium. What we did here was thoroughly analyze the plant microbiome, its pathobiome, that is the microbiome associated with the infected plants, and also the soil microbiome, and then strategically went into generating synthetic microbial communities. And as I speak, we are right now performing the field experiment to test this efficiency. Having said this, I would go back to the question, the prime question here. The reason why I selected this, well, there are several reasons. One, this is in the session called Ecology and Evolution of Microbiome. 2, because this is also a success story taken to the fields. And 3, because of the methodology that I'm going to talk about. So the question here was, can plant-associated microbiomes be modulated in toto to confer targeted benefits to the plant? And here we wanted to do microbiome modulation. The 2 stories here are related. The rhizomicrobiome transplantation was also modulation in toto, while what I'm going to talk about more is in terms of acclimatizing this microbiome to different stresses. I'll take a minute to explain this methodology to you. We started off with a soil which was very diverse. So we performed a coalescence wherein we brought together soil from different kinds of habitats just to increase the diversity. We let the plant grow and then started applying stress. In this case, I'm talking about salt stress, and the plant here is Vigna radiata. What we did was, after every passaging, we selected the Rizosphere from that plant and used it as an inoculum for the next passage. And this we kept on doing. As I speak, we are right now in the 28th passaging. The point here was we always grew them under sterile condition, in sterile soil, and then applied this as the inoculum. In the process, we also started increasing the level of stress, to the point that we saw that the controls were similar to the stress-infected plants inoculated with the microbiome. Now, what actually is happening? The microbiome is being engineered in two ways. Number one, there is a stress by which the microbiome, which is now enriched, is stress tolerant and is also conferring, the plant is selecting the microbiome, so it is actually conferring its benefit to the plant. But then there are also changes in the root exudation profile because of the stress. Hence, there would be a different kind of microbiome selected by the plant because of the stress. So these are the two forces acting here. What did we do? We performed this acclimatization over repeated cycles. We characterized the fungal and bacterial community to see how it was evolving. We quantified certain stress markers which were giving an indication as to how the microbiome application is helping the plant. To the best of my knowledge, we do not have a method in place right now to store the microbiome in its matrix, which is the soil here, to retain its functionality. There are several methods known to actually perform structural analysis, but we do not have a method in place to retain its function. So the only bypass we had then was to generate a culture bank of potent salt-tolerant strains. And we also performed certain experiments to understand the mechanics behind it. So we studied some volatiles which were involved in stress mitigation to be able to really target the active metabolites. We then went on to generate syncoms. How we generated syncoms is another story because we used certain strategies based on the functional assays. I'm not going to touch upon that. We tested the efficacy of the syncoms in plants, first under controlled conditions, and then we took it to the fields. This is just to show you a representative image. Wherever I say M, it is the microbiome inoculation. I want you to compare it with the salt-stressed, the S samples, and the M samples, which is the microbiome inoculated, like I said. Just to show you some more data, and I don't want to boggle you here, what I want you to see is just these 3 bars, and what I'm showing you here is the passaging. So basically the 7th passage to the 13th passage, and these 3 bars towards the end are the microbiome inoculated samples. I'm also showing you here certain stress markers. So for example, the MDA levels, which you can see upon application of microbiome went down. Also the membrane stability index, which was higher in the case of membrane microbiome inoculation. All that was good. We then wanted to understand how was the microbiome evolving along the different passages. What I'm showing you here is salt plus microbiome treated at the 5th passage and the 13th passage. And basically what I am coming to here is that the application of the microbiome was actually leading to rebiosis or replenishment of the lost diversity. Again, just to show you some changes in the diversity indices, again, I'm comparing here salt treated with the corresponding salt plus microbiome treated, and we could see this increase in diversity upon application of the microbiome. Like I said, then we had to actually revert to the culture-dependent approach of generating a culture bank. Here, this is a culture bank of stress-tolerant or salt-tolerant, in this case, microorganisms. We generated this and then took it to the controlled condition by generating syncoms. So this is what we saw in the laboratory. What I'm showing you here is that we could actually design syncoms which were mimicking the total microbiome application. So that gave us the confidence of then, first of course we looked at different kinds of parameters, membrane stability index. You see here we have the syncoms as well as the microbiome. In fact, some syncoms performed better than the microbiome. This was also the case when we looked at certain other markers like the total chlorophyll content, proline content, malondialdehyde levels. So this basically gave us the confidence of then taking this method to the fields. And that was the bigger question. So we found a field that was naturally saline and was not being used by the farmers. This is a field that we found in the Kanpur Dehat area. And without really showing you any data, I just wish to show you this picture which talks, I think it speaks volumes. This is the kind of soil that we had, and this is after the plants were grown. This is Vigna radiata. So to summarize the story, we could show that multi-passaging of the rhizosphere microbiome of Vigna radiata generated a stress acclimatized microbiome. This is the top-down approach of rhizosphere engineering. And herein, we could actually ramp up the salinity stress conditions and get a more tolerant microbiome to work with. The stress microbiome application led to enhanced plant growth attributes and reduced the stress marker levels. We could track the dynamics of the bacterial and fungal communities along the passaging. And the application of the SMCs enhanced plant growth attributes, including the yield under control and field conditions. Finally, microbiome-based rhizosphere engineering could serve as a potent strategy for mitigation of stresses. But this opened up several questions, and currently we're working with some. The first question is, can the microbiome members be tracked upon application? So we are right now working on identifying those signature members which can be tracked along the process so that we are sure of the robustness of the microbiome after application. The second question is, what is the spectrum of hosts for the engineered rhizosphere microbiome in mitigating stresses? Now what we're doing here is trying to apply this acclimatized microbiome, acclimatized for Vigna radiata, for different kinds of plants to check its efficiency. We're trying to see its efficiency in other legumes and non-legumes. We're also trying to see if this stress acclimatized microbiome, which is actually acclimatized for salinity stress tolerance, can be applied for various other stresses. Like the ongoing experiment right now is with drought tolerance, which is kind of related to the salinity stress. Then we're trying to understand what are the active ingredients responsible for their impact on the plant, not just for understanding the mechanistics, but also to see if this can be then applied into the field. And the bigger question is, how practical is the field application of microbiome-based rhizosphere engineering? I hope with this I have been able to throw some light on how soil microbiome and plant-associated microbiome can serve as a game changer for agriculture, because of the role of the microbiome not just in nutrient balance, in food security, in environmental sustainability and food safety, and how this can be used to address some of the SDGs. I've listed some, but it seems there's a lot more interactions that are happening place between the SDGs. And before I stop, I wish to say thank you to some of my collaborators. The work that I showed in the fields were performed with Dr. Y. Shivai from ICR-IRI, also with Dr. Ankita Sarkar from Banaras Hindu University. The work on the characterization of some of the microbiomes was done with Lars from DTU Denmark, and the story on volatiles, which we did to understand the mechanism, was done by Dr. Lor. This has received generous funding from Tata Sons, DBT, and DST. With that, I'm open to questions. Thank you all very much. And I have to say a big thank you to my working group.
Wow, I mean, this is just pure amazing. So I work on stool transplants, and there are so many parallels. The number one question I had in mind was, what will be the Cox postulates equivalent for this? So can you take away some of these rhizosphere transplants or alter it back to show that if we remove this, the soil health, the plant health deteriorates?
Yeah,
so something like that to add more. And also on the same topic, if you repeat this thing across multiple plants, across multiple sites, do you see a similar effect and all?
Yeah, so I think we're working on most of both the questions. In fact, we were wondering as to, or let me start with this. We had a tough time in designing the controls for this experiment when you do the acclimatization. What kind of controls really validate that it is the microbiome which is conferring the strategy? I'm not really sure of how to really go back and see if we can eliminate one or the other microbe to be sure that it is this conferring the particular fitness effect. But the idea is to not do that. So what we're looking at is to work with the microbiome in totality. So if we are actually removing a strain from that, we are actually disrupting the interactions, right? So as I speak, I'm thinking about the kind of experiments that could be designed to actually say for sure that it is an impact of this particular microbiome, but not microbe. And the second question was? Yeah, so to increase, to expand, right. So, while it's still in the preliminary stage, we are doing it, like I said, with Kajanus Kajan, we are doing it with tomato, and we're doing it with wheat, with some very promising result. But we have not gone to the field yet. These are experimental fields, not the farmer's field that we work with. So it seems that there is a universal stress acclimatized microbiome which is selected by the plant from that particular soil type, which is also affecting the others in a positive manner.
Just one follow-up question. So just like for stool FMT, we see a longevity of response.
Yes.
The response is there for 20 weeks, sometimes even 5 years, and there is a relapse.
Right.
Do you notice something here?
Yes. We call it priming. We call this particular phenomenon in this case as priming. You really see that longevity effect. Even if the actual members would probably have diminished or you're not able to track them, you can still see the effects down to the crop yield final stage.
Yeah, this is a very tough and interesting topic that you are doing it, especially for India. So a couple of things. When you collect samples from all over the India you showed, and mix it and then use it there, and each individual places have some dominant members and rare microbiome. Yes. We are all concentrating dominant microbes.
Yes.
And the rare microbes which you may able to culture, but sometimes you miss it because you have dominant microbes.
Yes.
So that is very important, looking into the rare microbiome that is actually doing a catalytic thing for the other thing. So, and how do you tackle that multiple dominant microbes you're bringing it and putting it in Jaipur and then making it done in an arid environment?
Very interesting question. I think all the plant microbiome people would look for a universal microbiome. They're trying to have a universal microbiome, and it does not work that way because the soil conditions are different. The idea here is to be able to identify the dominant ones, conferring suppressiveness, and if we could, you rightly said that it's the non-core which is equally important, and probably that is the one which is catalyzing it. But here the idea is to generate a culture bank of all these potent strains and the good microbiomes to see if there are some commonalities across the nation. So I know we are very far from having a universal microbiome in place.
That's right. But you are using the state-of-the-art molecular techniques to know what is the dominant microbes?
Yes.
Maybe,
and check its functionality.
And also you can simply grow to see in different arid conditions, for example, arid condition soil based, agar plate, for example. Yeah. Then you try to see which one is coming.
So the agar plate is a little far from the natural system. We try to do the plant assays wherein we check the suppressiveness with the soil extracts and to define it as a suppressive soil. And then we do the high-end omics technologies to study, to characterize what is in there actually.
Wonderful work and beautiful talk. I had a question about your serial passaging experiments for the acclimatization of these microbiomes. If I understood correctly, every time you passage it, you're using sterile soil. Yes. Then when you now have this acclimatized microbiome, you're going to use it in a setting where there is already a resident microbiome. Yes. So what's the rationale behind expecting that this selected microbiome will replace a resident colony?
Yeah, good. This is a microbiome, and the beauty of this approach lies in this fact that the plant is selecting its microbiome. We are letting nature act on its own course, and because the plant is selecting a microbiome, the robustness is not a question. So even when you apply it into a field or into this plant experiment wherein there is a resident microbiome, this community will have a strong colonization.
So I think you already answered many of these questions, or you posed them as questions in your final slide. So, so you were talking even in your previous answer that people want to see a universal microbiome. So instead, is there a sort of universal metabolome in the sense the kind of molecules that are being present there that are so it might be different species in the end, but the same kind of molecules that are ultimately improving plant health?
The reason for doing a metabolome analysis is just this. If we can actually chance upon a universal metabolome, it will make our life easy. then we're not going for the cell-based approaches, but the cell-free approaches of targeting. So that makes, partially, actually makes this life easy.
I have a question. So you were talking about two things in your introduction slides. One is the good microbiome of the rhizosphere and the bad microbiome of the rhizosphere. So having this consortia of microbiome that you're applying, can you comment on the effect of this over the bad microbiome? And extended question of this, probably, so I'm a phage biologist, and have you ever thought about using phages to mitigate the bad microbiome, please?
Yeah, so we have done studies, and in fact this was one of some of my earlier work that we have done, wherein we have shown that application of bioinoculants and these good microbiome are actually displacing the bad microbiome. When I say bad microbiome, I'm talking about the pathogens, the phytopathogens, right? So we have evidence for that. Yes, it is.
We'll just take one of the online questions at least. So the second one we'll go for. How is the rhizosphere microbiome different from that of the gut in terms of diversity and composition?
So there are a lot of differences. However, the four main phyla remain the same. So then there could be several other fluctuations and the genera would change. But these Firmicutes, Proteo, Acido, and Actino and Bacteroidetes remain the same.
And the diversity values are similar to what you find
diversity
in the gut?
Values are quite similar, yeah.
Okay. Yeah. So thank you very much for your very nice talk, ma'am.
So our next speaker is Dr. Deepa Agashi. So Deepa is an associate professor at NCBS Bangalore, and she received her PhD from University of Texas at Austin in 2009 and conducted postdoctoral research at Harvard University before returning to India. Her team uses experimental and bioinformatic approaches to understand the process and mechanism underlying adaptive evolution using insect and bacterial models. She received the Women Research Excellence Award from the SERB and the Faculty Excellence Award from Society of Molecular Biology and Evolution. She was the 2022 Vice President of American Society of Naturalists and was elected as a Fellow of the Indian Academy of Science in 2025. So we look forward to your talk, Deepa.
Thank you. I hope you can hear me in feedback. Right, I know tea is around the corner, so I'll try and keep you entertained. So I'm going to tell you about ecological drift in the flower beetle microbiome. And really the story, I'll focus on the flower beetle microbiome because that's something I know a bit more about. But the point I want to really get across is that while it's tempting to think of microbiomes as always being something that have adaptive value, I think non-adaptive stochastic processes can play a big role in what we observe as microbiomes. There's a lot of transient microbial members that come and go, and it's important to keep that in mind when we think of microbiomes and what they might be doing and how they might be evolving. Okay, so I think this is not new to most people in this audience, but host microbiome interactions are ubiquitous, they're diverse, and they're often important. This is just an example from a nice review paper showing all the different kinds of processes that microbes seem to be in charge of when it comes to associations with plants. And these kinds of effects have also really big impacts on global biogeochemical cycles, and you've heard about some of this work before in this session. But not all animals seem to need a microbiome, and the big question is why not? Here's again from a nice review, a series of work over the last couple decades has shown that a large number of animals may have strong associations with their microbiomes where there's a lot of beneficial symbionts. The example here is of ruminants. But there's also a bunch of other species that seem to have only very weak associations with their microbes, a lot of more transient species, and then a whole bunch of insects in particular, just because I think they've been well studied, which seem to also fall in this category where they don't seem to have any associations with their microbes. And the question is why? So we've kind of been swinging back and forth in the field as a whole, starting from this idea that, okay, animals are evolving on their own, largely sort of not associating with anybody else except in specific cases, to thinking, okay, everything is associated with some microbes or the other and depends on them. And then I think we sort of swinging a little bit back again, thinking, oh, actually a whole bunch of organisms don't seem to have microbes that they care about. And I'm going to tell you a few such case studies from our own work in the lab today. So here's some insights that we had when we first started working on insects in the wild. And I was keen on working on sort of wild species simply because I think that's where we are able to understand a lot more of what's going on compared to what happens when we bring things back in the lab. So we worked with a few butterflies and wanted to ask whether butterfly larvae who consume different host plant leaves, what kinds of microbiomes do they have? They're eating food which is not particularly great in terms of nutrition, so leaf is largely stuff that you cannot digest. What we find is that if you look at just simple 16S based classification of what microbes, what bacteria are there, the larvae largely sort of mirror what is present on the leaves, right? So these are butterflies that are eating citrus species. Here are some butterflies that eat other species. And in many of these cases where we sampled butterfly larvae from the wild, sampled the plants they were eating, they were kind of looking quite similar, suggesting that there isn't anything specific or any enrichment that's going on in the guts of these larvae as they grow up. We also find something similar with dragonflies. So this is showing you if we take a bunch of different dragonflies which we collect from the wild and we ask how many and which species of bacteria are present, what is their abundance, and we try and fit these null model analyses to these data for how often a particular microbe is present and at what abundance it's present. We find that most of these bacterial taxa fall into a category which is consistent with neutral presence or absence, right? So it's there, it's largely dispersal driven. It's really very, very few taxa that seem to show signatures of either positive or negative associations with the hosts. We also did some manipulative experiments with butterflies. So here's two species that feed on very, very toxic plants. And so we thought, okay, here's a case where surely the gut microbes have something to do with what the toxins that these species are consuming. But when we feed these larvae with antibiotics and we count how many days the individuals take to eclose, so that is to become adults, we find absolutely no change whether we treat with antibiotics, big dose, low dose, whatever. And we measure many, many different fitness parameters like this for the hosts, and we find the same thing, right? So here were several situations where we failed to find any microbe-host associations, and so a bunch of sort of plants went down the drain. And then we thought, okay, fine, we'll compromise and find this nice sweet spot between the wild and the laboratory. This is the red flower beetle. It's a pest species found globally, lives in grain warehouses, and it consumes wheat flour for the most part. It's really good at dealing with wheat flour. You might even have seen this in your own home sometimes if you leave some flour around for a very long time. It's a tiny, tiny pest. And what's nice about it is even though it specializes to some extent on wheat, it is able to eat many, many different kinds of flours, right? So it is a generalist in some sense. And we can bring it into the lab without sort of losing a lot of what's natural for the species. So it's been a pest with us for a very long time, at least since 2500 BC, where somebody has found these beetles in the grain that was buried with pharaohs in Egypt. So it's been a pest with us for a long time, and that this is now their natural habitat. And here because we can maintain these guys in the lab for relatively easily and pretty fast generation times, it's easy to start asking questions about the microbiome. Does it matter? Why? And in what way? So what we found in early work is that if we reduce the microbial load in the flower, I should mention that the beetles conduct their entire life cycle in the flower itself, right? So you have a box of flour which you give the beetles. They mate, they reproduce, they eat, they poop. Everything happens in the flower. So it's great in some sense because now the flower is the primary ecological niche through which everything is cycling. And because everything is cycling through there, if you deplete what's happening in the flower, you end up depleting what's happening in the beetle itself. So we can treat the flower with UV or antibiotics, and then we allow females to lay eggs in this flower, and we ask, do the females care at all whether there is the normal microbiome in the flower or not? And the answer is yes. So here's how many offspring are produced when females have access to normal flower. Here's how many offspring they have when they have access to flower which is treated with UV radiation or antibiotics. We also find the immune function, or a particular kind of immune function called immune priming, vaguely similar to immune memory that we know of in vertebrates. That also reduces. So here's the proportion of individuals that survive after being allowed exposure to a priming non-lethal dose of the bacteria. In the presence of normal flower microbes, the survival is follows this trajectory after some number of days after infection. But if you treat the flower with UV, you find that the survival crashes as much as it is without your option of priming. So it seems like there are fitness consequences, but none of these are really, really important in the sense that the beetles are perfectly fine in terms of survival and fitness without the microbes as well. The fitness is reduced, yes, but it's not lethal if you get rid of the microbes. The problem began when we started trying to repeat these experiments and we could not. Right? So this, as many of you will sympathize with, is horrifying. When you can't repeat your results that several people in the lab have painstakingly done over the years. You're okay, what's going on? These people have all left the lab. What are we doing? So several of us, this is early COVID times, we tried to repeat these results. Okay, no, it's not working out. So we said, okay, fine, maybe the microbiome itself is something weird and odd. Maybe it's changed. So we went back and collected all of the stocks that we had put in the freezer for some reason or the other. And we sequenced all of these stocks. And you do see, so every line here is a single female. Okay, this is all beetles of roughly the same age, which were stored in the lab freezer as part of different experiments, right? So they should be, you would expect they should be more or less the same thing. And clearly you see they're absolutely not, correct? So the microbiome is extremely variable and flips from 1 month to the other. You also notice that there's a lot of individual variation, right? So in some months, the individuals look all similar. They're dominated by some species of Enterococcus and what's identified as Escherichia, the orange one, which will become important later on. But in other months, there's a whole bunch of variation where different individuals have all kinds of rare taxa, which are lumped into these gray bars here, right? So there's not only individual variation, there's a lot of variation from months and years. And you can calculate the amount of individual variation using this taxonomic beta diversity. So positive values indicate very similar microbiomes across individuals from one single box. And negative values indicate very, very dissimilar microbiomes. And you can see even that value itself fluctuates over time. If we use qPCR to ask what is the total bacterial load relative to per unit beetle tissue, right? So we use an internal beetle primer for the qPCR. We find that the total bacterial load also changes. So how many bacteria each beetle is carrying is also fluctuating over time. What gives? The beetle microbiome varies also across months and years. Life stage, replicate boxes, parents and offspring. This is a total disaster. What are we doing here? So I'm not showing you all the data here. So okay, fine, there are lemons. Let's try and understand. Let's see if we can make some lemonade here. So why is the microbiome so unstable? Let's be a bit systematic about it. Here's where we can break down what might be happening in these flower boxes, right, into 3 different sort of stages. There's an inoculation or influx stage where the only sort of sources of microbes in the box of flower is the flower itself. It has some microbiome associated with it, the beetles, and potentially some contamination from handlers, air, etc. Right? So we did all the experiments to rule those out. So we're still left with some flower and some beetle influx. Then the microbes have to persist in flower for some time, right? Because there's this whole cycling through the beetle gut that's going on. And in that process, some microbes are going to die. Also, the flower itself, without the moisture being generated by the beetles, is a very inhospitable, dry environment. There's likely no growth of microbes that's going to be possible there. So probably there's some death that's happening just because the flower is very dry. And finally, there has to be colonization and assembly of the beetles themselves. And here we have to break it up into 2 different stages. The larvae are the eating machines, right? They're just eating and eating and eating and growing a lot. So they are the primary bags of microbes. And the adults live longer. They don't eat as much. They're reproducing a lot more. So potentially something has to go on here, which could be changing as they go through the pupal stage, where because there's complete metamorphosis, the entire gut is remodeled, right? There's very little microbial load in the pupae. So we decided we're going to explore all of this and understand what's going on and where this drift is going on. So what we find is if you look at just isolated larvae that we take in small groups, separate the larvae from the adults, and look at the total bacterial load of each of these over time. So this is the length of the duration of the larval stage versus the adult stage. Now notice this, the scale is weeks versus days here. So as I told you, adults live much longer than larvae. You find that in larvae, the load increases within about 6 to 9 days, right? So within a week, the larvae are really giant bags of microbes. In the adults, it takes a really long time for that increase in microbes to show up. And you need to remember this because this becomes important later on. The other thing to notice is that the pie charts show you what's going on with an average microbiome of the larvae and the adults at each of these stages. Again, you see that within a week, the larvae are full of this orange bacterium called Escherichia, which I suggested was important earlier, because it shows up whenever the loads are higher. But in adults, it takes a while for that to happen. So it takes about 3 weeks before the Escherichia is the dominant taxon in this group. So this becomes important. This suggests that colonization is much slower in adults compared to larvae. We also find that unlike larvae, when we take flour which has been used, that is pooped in by adults, there are some of these key bacteria missing. So here's fresh flour. It's full of very rare taxa. The load is very low, as you'd expect. Here is flour which has been used by adults only, right? And you can see there's hardly any difference with the fresh flour. But you take a bunch of larvae, put them in the flour, and you have a huge abundance and enrichment of these two bacteria, which we think are important for the beetles. If you now take some of these different kinds of flours, which are enriched with larvae or adults or controls, and ask how does the total bacterial load change when you introduce pupae, fresh, newly eclosed adults in there, you again see that the larval enriched flour allows for a huge increase in the load of beetles exposed to the larval enriched flour. But that doesn't happen for the adults. Also, the beta diversity, a measure of how much individual variation there is also really high when you have flour which has been enriched by adults, not so by larval enriched flours. So all of this suggested that something is very important and different between the larvae and the adults, and the question is why. So we go back to literature, and we had obviously seen before that the ecology of the flour beetle has been very well worked out before, and people had noted that there are these quinones, which are antimicrobial molecules released by the adult flour beetles in the flour itself. So these are released from these sting glands in the abdomen and 2 sting glands in the thorax as well. And we've known from assays, simple antimicrobial assays done on agar plates, that there is inhibition of bacterial growth due to these quinones. So when we isolate a bunch of different bacteria which we could culture from the beetle gut and ask how much inhibition do we observe, you see a wide range of inhibition, right? As you'd expect, that any chemical compound is going to inhibit some bacteria more than others, which is great. So this now suggested that there's some selective inhibition of specific taxa, and maybe this has to do with why we're seeing adults having this effect and not the larvae. And that's because the larvae don't have these sting glands, they don't produce quinones, but the adults do produce quinones. So it makes sense that because the adults are secreting quinones in the flour, the loads go down, and particularly Escherichia and Enterococcus seem to be affected more. So when we take little paper discs of quinones, which we extract from the adult adult beetles, and we put them in flour which has been conditioned by larvae, there is no impact, presumably because the larval enrichment of Escherichia is so strong and so high that even if you inhibit using quinones, there's still a very, very high enrichment compared to fresh flour. But then you take pupae and you put them in flour conditioned by adults, and you again recover this. So the quinones really seem to be having a big impact. And to finally sort of test this for sure, we decided to suppress quinone production using RNAi. So we designed treatments where we don't treat the adults with anything. They should have normal sting glands. We treat pupae with a control, which is our RNAi against GFP. This should have no effect because there's no GFP in these beetles at all. So they should be producing quinones as expected. And then we take this one gene, which is part of the quinone synthesis pathway. This is shown in previous literature. And when we suppress the production of this gene, expression of this gene, we should be getting no quinones. We now put these beetles when they close into flour and allow that flour to sort of get enriched or not with whatever microbes are there. And we then sample the microbes and look at the microbial loads to ask what happens to adults which have been exposed to these conditions. If you look at if you dissect these beetles, you find that indeed, as expected, the untreated beetles have normal sting glands. The control GFP treated beetles also have normal sting glands, but then our RNAi beetles for the quinone pathway are missing the sting glands. So fine, everything is working as expected. And then we do see that the bacterial loads are also working as expected. with the GD39 suppression, leading to a much higher bacterial load in the flower compared to what we see in the other treatments. So what we're suggesting then is that there is a lot of stochasticity that's arising because of production of quinones by these adults. So this is then the working model. The beetles are producing feces and eggs. I haven't shown you the data today, but we've looked at the egg microbiome, and we don't find a very high load or a significant enrichment of specific microbes. So we don't think this is a big source of inoculum. We've also done experiments showing that if you introduce two flour enriched with specific microbes, if you introduce quinones there, there's a lot of death that happens due to the quinones, but also death that happens due to the dryness of the flour. And then what we've done some work on is to find out whether there are any priority effects or metabolic differences between microbes that decide which of the microbes are able to colonize better or not, separate from the effect of quinones. So with all of this, I still haven't answered ultimately how is ecological drift generated in our populations. And the last clue came from when we looked at what's happening through a normal cycle of maintaining stocks in the lab, right? So you have to change the flour that we give to these beetles on a regular basis, otherwise it's going to become nasty and low nutrition and it's not good. So the blue arrows here indicate maintenance cycles where we add fresh flour to the box and change out the old flour. And you see that every time we add fresh flour, you have a drop in the total bacterial load. This makes sense because we're adding fresh flour, right? But what's also important then is whenever there are larvae around, there's a higher proportion of Escherichia. Wherever there are adults around, there is no Escherichia, right? So what we think is going on is in our normal process of maintaining nice, healthy, clean populations in the laboratory, it takes about a week for adults to lay eggs. We remove the adults. The eggs develop as larvae. It takes them about 3 weeks to get to pupal stage. And then we let them mature for 1 to 2 weeks. And then we take these adults and start the next generation. That's the cycle we follow in the lab. Now what happens is we think drift is generated in two places here. Here, we have at this stage, we have a flower change after about 1 week. What happens is that during that flower change, most of these individuals, which were larvae, have now become pupae. And the pupae don't contribute anything to the microbiome. They're more or less sterile themselves inside, right? Which means that now there's fresh flour, and there are pupae, no larvae around, to really introduce the microbes that should be enriched in the microbiome. What also happens is the adult age at initiation, the next generation, is 1 to 2 weeks. This is before the adult microbiome stabilizes. It takes 3 to 4 weeks for that to happen, which means that every time we have a new generation started off our stocks, we're starting from base 0, right? Then everything is super stochastic, and so we think this is why there's a lot of drift showing up in our microbes. So to summarize what I've shown you, drift can shape microbial assembly and perhaps the evolution of host microbial interactions. Even under controlled laboratory conditions, we find that microbiomes are super unstable. We suggest that the sources of such drift are many and they can vary a lot. And finally, ecological drift of this sort may potentially inhibit the formation of strong host microbial associations. This may not be true only for our model system, our species, and so on. We suspect it might be true in many, many cases where we don't observe very, very strong obligate relationships between hosts and their microbes. And I think this is important to keep in mind to be able to understand how host microbial relationships are established. In my mind, it's just super surprising that any of these obligate relationships exist at all, because it just seems so difficult. You know, everything is going against you. You have to find your partner, you have to set up a relationship, they have to depend on you, you have to depend on them. Hard. Okay, so I'll end by thanking all the people who've been important for this work. Give a shout out especially to Pratibha. She's a senior postdoc in the lab who's led charge of all of this work in the last 2 to 3 years. As we started out from not understanding what's going on to sort of figuring out, okay, there's some explanation for what's going on. She was helped by a lot of people in the lab over the years. And Pratibha was supposed to be at this meeting, but unfortunately had to cancel at the last minute. I'm sad that she's not here because she would have really liked listening to all these talks. Also thank the sequencing facility at NCBS, which has been absolutely fantastic, helped us find hacks to also reduce how much money we have to pay for the sequencing. And the TIGS insect facility also on our campus where we can do all the RNAi work. The sources of funding, and thank you for your attention. I'm happy to take questions.
Thank you very much, Deepa. So if I can ask first question. So Ezekiela, which kind of microbe is it? Like, is it classification or taxonomy?
Kind of in the Erwiniaceae, Enterobacteriaceae clade.
Okay.
But it has shifted its classification as databases get updated.
Yeah.
And the most annoying thing is we've not been able to culture it. So we've been trying to culture this stupid bacterium for 2 years.
But it's like you guys are not able to culture it, or it has not been cultured till now?
No, so there is one species of Ezekiela which was isolated from a mirrored bug, and we have that from a culture collection. And that's what we've been trying to use here, but it's not a good colonizer of our beetles.
Does it have a complete genome to some extent?
There is a complete genome, yeah. So we're trying to get the genome for our bug, but...
Okay, so, yeah.
Thank you for the interesting talk. Maybe I missed it. What is the type of quinone that is produced by this adult beetle, and have you tried to quantify what is the level of quinone produced?
Yeah, so we have quantified it in previous work, and other people have also done the same many years ago. It's ethyl benzoquinone and methyl benzoquinone primarily. I think EBL is a larger proportion.
It's a very good talk. It's a very good talk and economically important as well. So is that quinone is produced by different, what do you call it, not species, it is strains of this beetle, right? It can vary from beetle to beetle, right? Yes?
So yes, it can vary, but it also actually varies a lot based on age and sex and crowding.
And also the characteristics are different, or it is consistently producing the same type of quinone, or its derivatives?
It's primarily the same type. The composition doesn't change very much across age and sex, but it does change between species. So closely related species, many of them produce quinones, but slightly varying in their composition.
In practice, how do you envision to have this quinone being added into the flower? So,
oh, we should not. It's a carcinogen. It's terrible to add it to the flower. No. We should not be doing it. That's actually partly what makes the flower unpalatable for humans when you have a serious infestation. It's also awfully stinky.
But just to quickly say, can it be a marker for knowing that there are beetles or infestation?
To some extent. The problem is that these are very volatile compounds, and so after a while it's very hard to detect them. Okay. And actually even right now quantifying the amounts from flour is a big challenge.
Hi, Deepa, this is me here, Yamini. Okay, so I think I have two questions, but I'm trying to figure out my second question. But first question is that did you, maybe I missed it, but did you also sample the flower and the microbiota of the flower, and then with every change in inoculum, did you compare how the microbiome has changed from flower coming from a company versus second part number or whatever on those lines?
We haven't looked at different companies, but whenever we've tested flower,
as in the same company, different lot numbers, sometimes that could be also problematic.
Yeah,
do you treat your flower before actually putting your beetle in it?
Yeah, so we usually sterilize our flower at -80 before, overnight before we use any of the flower. But in general, the flower, if it's like flower in good condition, it's fine. It doesn't have a very high load.
So it doesn't have any microbiome community in the first place.
We haven't done a systematic analysis of this, but over the years, whatever we've sampled doesn't show very big differences.
Okay. So my second part is also that a couple of, as in I don't work on this similar lines, but I was listening to a couple of talks where in rat they do something similar, but they have a rat microbiome completely like zero microbiome, and then they infest it with certain sort of environment and see how much the rat has ingested, or how the microbiome of that rat has evolved over a period of time. So what I'm seeing here is that your beetle microbiome is changing over a period of time. So how do you, I'm trying to understand the overall impact of this, what is the upper
Why should anybody
question?
Care?
Yes.
Yeah,
sorry.
So I think, like I said earlier, it shows really clearly, I think, that there are many sources of drift in the assembly of microbes, of microbiomes associated with any organism probably. And it's important to keep those in mind and look for them and these sources of stochasticity, because they can come from your nice protocols in place to keep your populations healthy. So I think it's just that I suspect that this is not uncommon. By the way, for this pest, I think what we see in the lab is should be mirrored very clearly in its natural environment as well. Because in a flower warehouse, there's a random influx of flour. People will spray pesticides. So same kinds of processes are going to keep happening. There's colonization, there's extinction locally, and then there's recolonization from other sources of beetles from other parts. So I suspect these dynamics are very, very frequent in at least this species. And I started working with this because I'm like, okay, this is simple, we can manipulate this, right? And turns out it's not. So I don't think it's very rare.
So I had a question about the last point you made, which is quite interesting in terms of ecological drift versus host microbiome interaction. Is there a way to quantify each? Maybe you've already done that so that we can just look at it in a plot to say, is there an inverse relation? Or a more detail of that question is, are there like ecological drift quantified on a per species level and then also host microbial interaction or host species interaction done so that we can look at it?
Yeah, I see what you're saying, but I don't think we have the data to be able to do that. Partly because people who study microbial assembly in these kinds of habitats or species which are non-obligate associations are not working with species that have obligate associations. Where there's an obligate association, there's no question of asking how much drift, etc. Right? There is an association, and in some sense, just in principle or logically, you would expect that if there is a strong obligate association, usually it goes along with things like maternal transfer of the symbionts that are important, or deposition of the symbiont just outside in the environment that can be taken up quickly by the next generation. So the point is that the reduction in drift it goes hand in hand with the evolution of an obligate relationship. You can't have one in the presence of the other. So so I see that it's important to quantify, but I think it's hard to do.
Thanks.
Any other question? Okay, yeah. You can take one more.
Hi, so I just had a quick question. Since we're in the age of MAGs and metagenomes and everything, have you considered, since there is at least one isolate that is already present, to create a MAG out of this? Because it's quite interesting that it's only the Ezekiela population is only present in one life portion of the life cycle, and if it's an enterobacterial, then in theory it shouldn't be really too beneficial for the host organism. So
yeah, I don't know. So yes, we've tried that, but what I am missing at the moment is somebody in my lab who can assemble MAGs. Like you read the papers, it looks doable, and then we try and do it, and so if you have any ideas, please, I'm all ears. Thanks a lot.
Thank you so much. Okay, thank you very much. Thank you very much, Deepa.
Yeah.
Brett, can you come? So I think we have 30 minutes break. Okay.
get started. I mean,
Welcome back to session 3 on environmental microbiomes. This session will be chaired by Professor Shankar Manoharan and Dr. Vijaya Raghavan. I'll briefly introduce them. Professor Shankar Manoharan leads the microbial physiology laboratory at IIT Jodhpur, and his group is currently studying the effect of food system changes on the gut microbiota of vulnerable tribal populations in India. They also look at microbiota of biocrust, unique communities that engineer arid zone ecosystems with the aim of conserving, restoring, and tapping into the biosynthetic potential of these important desert communities. Dr. Vijaya Raghavan is a scientist at the Marine Biotechnology Division at NIOT, National Institute of Ocean Technology, in Chennai since May 2012. He has undertaken projects such as microbial community profiling of deep sea samples, functional metagenomics for bioprospection, and the development of bioinformatics pipelines for next generation sequencing. I ask you to introduce the speaker.
So, good evening all. We are looking forward to some interesting talks on the environmental microbiome session here. We have a list of eminent speakers lined up for the evening. To start with, we have amongst us Dr. Jennifer Petrich. She is a senior staff scientist at the Lawrence Livermore National Laboratory and a leading expert in environmental microbiology and biogeochemistry. Dr. Petrich earned her master's in forest science from the Yale School of Environment. She completed her PhD at the University of California, Berkeley, focusing on how fluctuating redox conditions influence the microbial ecology. Her focus is on soil carbon cycling, microbial ecology, and the use of advanced isotopic and imaging techniques to study microbial interactions with the environment. Her work involves developing isotopic and imaging tools to study carbon dynamics and investigating plant-based solutions for carbon sequestration, such as deep-rooted switchgrass. She has received several prestigious honors, including the Ernest Orlando Lawrence Award in the year 2021 and the 2019 Geochemical Society Endowed Biogeochemistry Lecture. So it's over to you, ma'am.
Thanks so much for having me. It's a great honor to be here and to talk about the work that we do as part of a project we call Microbes Persist. This is team science, which is maybe a little bit new to you. This is something that's a hallmark of the Department of Energy in the United States. And sometimes when people hear about the national labs or the Department of Energy, they think about the weapons program or national security. And I like to argue that the work we do is part of national security because food security, climate security is national security. So that's why we do this work. And I want to start by giving credit to my colleagues, including Jill, who's with us, and all of the young trainees who did the work that I'm going to tell you about. We really work together as a team. Without these folks, I wouldn't be standing with you. So I mentioned climate. Climate affects microorganisms. It's affecting their effects on ecosystems and the benefits that we derived from those ecosystems. And currently the way we understand where those ecosystems are headed in a warmer, drier, higher CO2 world is wrong, to be honest. And in part it's wrong because we've got the microbiology left out of the equation. So if we want to get some of the right answers, whether it's how soils are going to respond to warming, how increased amounts of litter fall from trees and grasses are going to affect carbon cycling, we have to have the microbial functionality included in our models. So that's one of the big reasons that I and our team work on what we work on. Most of my talk, though, I'm going to focus on soil carbon, and I want to give you a little bit of backdrop on why. We're practicing agriculture on something like 40% of the land surface of our planet. It's a huge amount, the terrestrial land surface. And it's a huge amount of destruction, to be honest, in terms of the soil carbon stocks. Soil carbon is the greatest stock it's greater than what we have in the biosphere and the atmosphere combined. And those carbon stocks are easy to destroy and to lose when you till the soil. And so we have something like 133 gigatons, those are billions of tons, that have released to the atmosphere. It's been released as CO2, so it's exacerbating our CO2 problem in our atmosphere that we already have. Some of it is actually drained down riverways, and so it's caused its erosion that's ended up in various marine systems. But the point is we've got a real debt to repay to our soils, because that carbon in soils is typically there as organic matter. And what is organic matter but a sponge? A sponge for water, a sponge for nutrients, a sponge for microorganisms to live in. And that carbon has so many benefits to the plants that we want to grow, whether it's in our agricultural systems or in natural systems. So restoring that carbon is it's actually sort of half my work. I'm not going to talk about it today. But I want to first talk about the fundamentals of how does that carbon get there in the first place? How does it persist? What is the microbial role in those dynamics? Well, the first thing I want you to know, if you don't already, is that microorganisms make up something like 50% of soil organic matter. They're dead bodies. They're necromass. Really only about 5% are alive. And so here you're looking at just an SEM of some rod-shaped bacteria. I don't know exactly what they are, and some arbuscular mycorrhizae. And you can just kind of start to envision how that biomass accumulates over time. But most of it is, as I say, is decaying. So we need to understand both how microorganisms live and the effects that they have while they're alive, but also how they die and the effects of their death and the materials that are then distributed throughout the food web. So the hypothesis at the writ large of our project is essentially that the ecophysiological traits, that how organisms live and die and what they're made of, all of that together at some level should be a predictor of soil carbon dynamics. And long ago, when we started our project, it's been about 7 years, I think, we put together this great big long list, and I'm sure you can't read it in the back, no worries. But essentially, it was a number of different microbial traits that we thought we could read out of our omics data, whether it's proteomics or metabolites or metagenomics, metatranscriptomes. And it was everything from biophysical traits, things how large an organism is, how fast it grows, its demography, to things like its stress response or emergent traits that have to do with its carbon use efficiency. All of those we thought we might be able to have a way of getting at the whole community metrics of traits through our newfound ability to do omics of whole communities. The answer is we're not doing well on answering that kind of question. This is just one paper that's done a synthesis of the literature. They looked at protein coding genes and the biogeochemical function of that we assume that gene carries out, and looked for patterns in the many hundreds of papers that have been collected. And they found a lot of assumptions and very little statistical support. We're not doing well at using microbial data to predict ecosystem biogeochemistry. Well, that's a challenge. That's something we need to fix, isn't it? And I think part of the problem is we're making, forgive me for saying it so strongly, but we're making the wrong measurements. And I say this, and forgive me because I'm speaking to an audience that has different reasons for doing microbiology. But if our interest is in predicting the biogeochemistry of the system at that moment, how much nitrous oxide is being emitted, how much methane, how much sulfur is being oxidized, we need to actually understand the organisms who are active at that moment. Because everyone else who might be dormant, or it might even be relic DNA, or maybe a spore, that DNA is making for a blurry picture. So I think it's important that we have ways to understand who's active, who's growing, who's dormant, who's deceased, and those different metabolic states, understanding how they shape the environment. So the two main tools that I'm going to be talking about today where we try to get at that active microbiome are metatranscriptomics, or characterizing a whole community RNA gene expression, and stable isotope probing, or SIP. And I'm going to assume you know what meta-T is, and I want to spend a little time on isotope probing because I don't think anyone else has talked about it today. So there are many ways to do stable isotope probing, but I'm going to talk about one particular type we call density gradient. The idea here is we introduce stable isotopes that are rare into the community, a whole intact community, in the field if we can. It might be 18O labeled water, which is really just water where the oxygens have been, the 16 oxygens have been replaced with 18, slightly heavier. We might be growing a plant in a chamber filled with the expensive kind of CO2, the 13 CO2. Then the plant takes it up just as it normally would, photosynthesis, we get it into the root exudates, we get it into the microbiome, and the microbiome doesn't know the difference. Once that heavy isotope is incorporated into the DNA of organisms that are growing and dividing, the replication, we're actually able to extract that DNA just as we normally would, and then we put it in a centrifuge. We go home for the weekend, 5 days later we're back, and that DNA has actually separated based on the density. So the heavier DNA actually migrates further in a cesium chloride gradient. And I always like to joke that we never see it, that's good, right? 2 bands. This is with a fully labeled 13C bacillus and a fully unlabeled culture as well. What we really get is a smear of lighter DNA to heavier DNA. We have to account for the GC effects, et cetera, but we take into all of that account, and we're able to have the mathematical equations where we normalize to the abundance of the organisms, and we get to quantifiable rates of growth per taxon without having to culture anyone. I'm terrible at culturing. So this is my way of getting at it. And we have many, many different ways that we run these experiments, whether we grow plants in the greenhouse, in chambers, we can do this in the field, we can couple it with nanosims imaging. Right, our latest invention, as it were, with the help of the JGI, was to create a series of robots that take the really tedious steps of this technique and make it so that we can go home at night and the robot keeps going. And so we're now running thousands of samples. The JGI has the same system, and you can apply to work with them, and they'll do it for you as well. Now, why is it's so important to be able to query what's going on with wild microbes? And I want to give you one illustration. This, I'm not going to tell you all the details, but this is a fairly complicated experiment where we had a plant. It was inoculated with a couple of different kinds of mycorrhizae. We had drought treatments. We had a lot of different treatments. And my then postdoc, Rachel Hestrin, did the initial community analysis and made this ordination. And this is one of those grad student crying moments because there's no difference. There's no statistical difference in the patterns of the community. But this is the whole community. And when we looked at just who was actively incorporating the isotope label, then it became very clear that our treatments had had an effect. The drought, the mycorrhizae, the different time points had had a strong effect on who was active. So that is, for me, one of the main motivators for why this technique is so useful. Another one is that this is something that we've seen over and over again in individual data sets, but my colleague Megan Foley synthesized recently in a paper. She took to get all the different papers where she could find stable isotope probing having been used for growth with 18O CYP. And she saw kind of a subtle but interesting phylogenetic pattern that different organisms tend to grow more. Okay, that's believable. But when she plotted all the data, she showed that there's really no relationship between abundance and relative growth rate. And I see this in study after study after study. The organisms who are abundant are not necessarily growing, replicating, dividing quickly. And that's important if you think about probably assumptions that you've made for a long time. One last thing that I want to tell you about as a cautionary tale is that I think we make assumptions about how lab cultures predict their wild cousins, right? You're sitting there thinking, yeah, I do that all the time. What's wrong with that? Well, here's an example where we looked at traits that we thought would be important for predicting growth rate, and that's simply copy number. The more copies you have of your RNA, the more you're able to ramp up quickly and grow, right? That's an assumption that I think is well held, and it's well supported by pure cultures. When we looked at the closest cousin that we could find in a wild setting, the pattern doesn't hold. Essentially, copy number does not predict growth rate, except for when we feed the organisms. So if we take a wild soil and we add glucose or add ammonium, add a bunch of resource, all of a sudden we do see that pattern again. Copy number and growth do have a positive correlation. But under natural conditions in soil, resources like that are not available. It is a desert most of the time in soil, unless a root is coming by. And so I think it's important, again, to just recognize that while what we find in the laboratory is important for telling us about the potential of organisms, it's not necessarily always being realized in the real world. So this is just those three caveats about in situ behavior not being predicted always by lab cultures. The responsive active taxa are in a relatively small subset. Sometimes as little as 8% of the community is actually growing. And taxon abundance doesn't necessarily predict activity. Okay, so back to my main question. What traits of living and dead microbes predict soil carbon cycling? So I'm going to talk about two different sets of experiments today. The first one is about Rizosphere. And I'm so glad you actually know what a Rizosphere is already. We had a lovely introduction to that earlier. Thank you. I was going to tell you a bit more, but I'll just say briefly that Rizosphere is, I just think of it as the hotbed of activity in soil. It's where you have much, much higher growth. You've got a lot of predation, though. At the same time, you've got a lot of phage. And we see a ton more enzyme activity. I could go on and on. As compared to what we call a detritosphere. A detritosphere is when that root is starting to decay. It's no longer exuding all of those wonderful compounds that microorganisms are responding to. So that's a very different resource environment. It's a lignocellulose-driven economy as opposed to a small molecular weight metabolite economy. And then compare that again to what we often call the bulk soil, the desert. You're not in the gut anymore of the plant. You're out in space where resources are far and few between, and there's very little microbial movement to cause interaction between organisms. So we set up a number of experiments where we looked at these things and combined it with isotope tracing. And the first person to really put together metagenome assembled genomes from this kind of work was Evan Starr, who was working with Jill. And I think what we were most excited about in this case was not that he put together a bunch of bacterial genomes, but that he found eukarya and that he found phage. And I'm sure that if Evan were to do this again today, we would still be scrolling. There would be a long, long list. But the organisms who were actively growing using our stable isotope probing techniques, we're actively being targeted by lots and lots of phage. So that suggests something just right there, doesn't it, about the activity that's going on in the rhizosphere. So if we look at what traits are causing changes in the carbon cycle in the rhizosphere, and we compare it to that decaying root or detritosphere, there are significant differences. And the fast bacterial growth that we see in the living root is accompanied by high carbon use efficiency, high amounts of extracellular polymeric substances, or EPS, production, and all of those are contributing to the formation of persistent organic matter that's sorbed onto mineral surfaces. Whereas in contrast, in that decaying root zone, we've got other traits, things that have to do with exoenzymes, and it tends to be a more slow-growing fungal-dominated community. Interestingly enough, when we drought the whole thing, the rhizosphere starts acting like that decaying root, and I think that's largely because we lose the water films that allow connection to happen. So we found there were a number of traits that were quite predictive. We then started to use metatranscriptomes to try and tell us at a more high-resolution temporal scale, what are these microbes thinking before the communities have even changed? And to do that, excuse me, we took RNA, depleted it. Sorry, I shouldn't say we did, JGI did. I'm looking at Tanya. I want to give full credit. The JGI helped us deplete the ribosomal RNA so that we have a not fully mRNA pool, but much more so. And we then sequenced those transcripts and classified them, mapped them back onto some of those mags that our colleague Evan had produced. And that allowed us to characterize essentially different guilds of organisms, but separate them by those habitats, the rhizosphere, the detritosphere. And so Aaron Nuccio published this work focusing mostly on the cazymes, or carbohydrate-dependent enzymes, where we're looking at all kinds of different carbohydrates as they're being broken down. And what she showed, it's quite a complex figure here, but you have differences in those guilds of organisms, both in time and also with the types of substrates that they are targeting. Interestingly enough, when she mapped these back to mags, the best mags to map to were CYP mags, so the enriched organisms were recruiting the most transcripts. We see rapid phylogenetic succession. So this experiment actually only lasted about 3 weeks, and the transcripts were changing 3 or 4 times over within that period. If we had looked just at the mags or just at 16S, there might have just been an inclination of community change. So there's clearly a lot of wheels spinning within the system that we wouldn't see if we're only looking at the DNA pool. Subsequently, other colleagues, Ella Sirajki and others, came along and took that same data set of gene transcripts, and it published a number of additional papers focusing on nitrogen cycling genes, on proteases. And Evan comes back into the story because Evan realized that there was actually an RNA virus story to be told here. Again, taking those sequences from that metatranscriptome, he discovered an abundance of RNA viruses. I've talked to a couple of people here today about them. We know nowhere near enough about soil RNA viruses, but he found that the patterns of these viruses was changing both in time and space, just as those transcripts had been. Right, so the second story that I'll tell you about is a particularly important moment in the ecosystem that I live in Northern California. I'm sure there are places in India that have similar effects. When you have a very, very dry soil that all of a sudden has a moment of rainfall, we call that a wet up. In California, it happens because we have a Mediterranean climate, and we go all summer long without a drop. And then there's this sort of magic moment in late August or September when it starts to rain, and you can smell it, right? And you can literally almost feel the soil exhaling, and it is. We call that the birch effect. The system can lose something like 10% of the net ecosystem carbon production from the entire year. It can lose that much carbon in a day through CO2 efflux from the soil, just in that first moment. So we've recreated that a number of times in the laboratory by collecting soil during that dry period at the end of August, bringing it to the lab, and then adding some 18-0 expensive rainfall to see how microorganisms respond. And respond they do. So there's an incredible difference in the starting community at 3 hours to what we see at 24 hours and then 72 and such. In that first week, there's just a bloom of growth of a limited number of phylogenies. There's actually even more death. Everyone dies. If we looked at all the different phylotypes, everyone is succumbing to a significant amount of mortality. And it happens really quickly within the first few hours. What's causing that mortality? This is something that folks looked at for a long time. They thought it was maybe osmotic shock, et cetera. We've learned it's actually probably not. But one thought was phage. So we looked at the phage sequences, and I want to give credit to Alexa Nicholas who did this, over that sequence of post-wet up. And what she showed is that initially dry soil is a really rich reservoir of viral sequences that actually they seem to have accumulated over the course of that long summer drought. But in that 24 hours post-wet up, we see a massive degradation of viruses. So it's not just the microbes that are dying. I don't know what we call it when viruses degrade. We're not allowed to say they die, right? They're going. And by 1 week, that biomass has come back up again. When we did some calculations to look at how much that 1 week response might have caused of the original Birch effect, we get a number that could be anywhere as high as 46% of the carbon turnover that was caused initially by the wet up could be driven by phage. That's an upper estimate, but I'll tell you in the oceans, it's easily 50 to 100% of the biomass turned over every single day by phage. So this is a particularly dramatic moment in soil, but we've been a long time wondering how much of carbon turnover is driven by phage. So even to know that just for this kind of one moment felt like a big step forward. And when we've done some modeling to actually try and predict the amount of CO2 that's lost from the soil, we find that taking both the community growth rate of using STIP and the active organisms and the phage degradation, putting that together, we get an amazingly good correlation between the production of carbon dioxide, the carbon that's being lost from the system, and that community growth rate. Now, our colleagues, and this was work done in Jill's group, Pete Chukran and Ella Shirajki looked at mags of those organisms that were growing and tried to find, is there anything in the genomes that would be a predictor of whether they were growing early or a little bit later? It was difficult to find clear patterns. We found some evidence that chitin is being upregulated in those genomic capacity to break down chitin is being upregulated in those early days. But I'll be honest that the story is not as clear as I would have hoped it might have been. But interestingly, what is clear, and this is data that Pete just published, is that there are patterns of codon usage, and there are patterns in response to that wet up. So we can categorize, based on the gene transcripts, who is an early responder to the wet up versus sort of a mid and a late responder, and who was what we call sensitive, who just was knocked out, who stopped transcribing at all. And so these colors here over time are meant to reflect those different categories, and they have remarkable differences in the way that they're using codons. So they're synonymous codons, if you're not know, are ways that encode for the same amino acid but with different bases. But the different bases have different ATP cost. So you can make a codon cheaply and you can make it in a more expensive way. But what we're seeing is that those early responders tended to use the cheaper codons. And that is maintained only when the peptide sequence that codon is coding for is not impacted. We actually looked at this at a phylogenetic level in our mags. We see that, as is not surprising, the proteobacteria are the sort of key early responders. And in those mags, that early transcriptional response was also associated with higher levels of this codon bias. So I'll be honest, codon usage wasn't on that list that I had at the beginning of my talk of different traits. It's not what I would have expected. But it actually turns out that these sort of ubiquitous genome traits like genome size, ribosomal copy number to some extent, and codon usage are some of the best predictors of carbon patterns writ large. This is data from the entire United States from about 30 different sites, neon sites. And what Pete's shown here is that the patterns of codon usage and codon frequency actually correlate with things like carbon to nitrogen ratio, really gross level characteristics of soils, suggesting that the carbon availability may actually be driving over evolutionary time the genomic characteristics in the soil. So just to end, thinking a little bit more again about the microbes that matter for predicting biogeochemistry, I think that I want to leave you with the idea that the genomic capacities, the gene expression, the interactions of wild microbes are really more likely to predict ecosystem scale effects than what we see in lab cultures. And we tend to extract their whole DNA, their whole RNA, their peptides, but that can include a lot of elements that make the picture blurry. So if we really want to predict what's going on in the moment, we need to find a technique that really tells us about those active organisms, who's growing and who's dying. We also tend to sample at the bulk scale. I like to joke that we go out with a shovel when we really should be taking tweezers. And I say that because the rhizosphere is different than a detritosphere that's different than a bulk soil, is different than the inside and the outside of a small aggregate. Just as in our gut, those subtle differences have an enormous difference in microbiome. These patterns in time and space really matter. And if we smear across them, we're less likely to see predictive patterns. And then I'll say the final thing that I think we've seen is that studying one domain, or let's call it kingdom in quotes, at a time is, again, not helping us to be able to predict what the ecosystem is doing. If we don't have the predators, if we don't have the prey involved, if we don't have multiple trophic levels, we don't get the full picture. So a thank you to my colleagues and the funding from mostly Department of Energy colleagues at JGI who did a lot of the sequencing, and happy to answer questions.
Thank you, Dr. Petrich. So, any questions from the audience?
A very great talk, and especially, dead and live is a big issue for monitoring microbes in any given environment. So, your stable isotope studies, can the spores, which are actually the dormant stage, okay, can the spores uptake the stable isotopes? And so when you measure it?
We don't have any evidence that they do. The spore coat is actually quite impermeable, so we've actually done some nanosims imaging to try and see if they're even just absorbing the isotopes that we add, but we don't see it. Yeah.
It's not uptaking, is it?
Sorry?
Is it uptaking?
No.
No. Okay.
No.
So then I cannot use it for me. Okay, thanks.
Sorry. Yeah, no. Yeah.
Thank you. I think we have a question there.
Jennifer, super cool talk actually. So I was wondering, you were telling about few active phages that came out after the rain. And do you know whether they are lytic or lysogenic phages system?
Okay, so it's a great question. We've got a whole series of projects right now where we're trying to take phage sequences from CIP experiments collected in many different ways. The extracellular DNA, the metagenomic sequences, which include prophages, and then anything from a true virome, which is a filtered virome. And those, there's a paper on this, so I'm not the first to tell you this, but the Venn diagram has got relatively little overlap. It's quite frustrating, to be honest. that each different pool is giving us a different set of phage. So that suggests that we're getting a mixture, and that in our metagenomes we're likely getting more prophages in the lysogenic state, but it's complicated, and I think that we don't fully understand what we're getting.
Thank you. Any other questions from the audience here? If there are no questions here, I think we can take up the questions from Slido. So it is back up from the hibernation mode. We can answer the second question. Given the huge biomass of dead microbes, is it safe to say that there's a lot of memory in the soil and the community trajectory is deeply influenced by it?
That's an interesting way of framing it. The biomass, if it's dead, it's not bio. it's not, the DNA is certainly there as a part of the soil organic matter pool. Do I think that genetic material is helping to inform further generations through some sort of nucleic acid transfer? I don't have evidence of that. Yeah, I'll think about it, but I actually don't think about that dead biomass as shaping the trajectory of who's there in the next generation, other than the fact that organic matter, as I say, is a sponge, is important for holding water and nutrients and all of those things. Yeah.
Thank you. Thank you, Dr. Petridge.
Okay, good evening, ladies and gentlemen. I'd like to thank the organizers for curating such a nice lineup of speakers on behalf of all of us. The next speaker is Dr. Yamini Jangir. She is He's a senior investigator at the National Center for Biotechnology Information, National Library of Medicine. Is he logged in? Fantastic. National Institutes of Health. He's a leading expert in computational biology and evolutionary genomics, specializing in comparative genomic analysis, phylogenetics, and the study of major evolutionary transitions. Dr. Kunin received his Master of Science and PhD in molecular biology from the Moscow State University. He has been a pivotal figure in the study of major transitions in the evolution of life, such as the origin of eukaryotes and the evolution of eukaryotic signaling and developmental pathways. Since joining NCBI in 1991, Dr. Kunin has contributed to genome annotation methods and mathematical modeling of genome evolution. He's an advocate of the extended evolutionary synthesis and has authored influential works, including The Logic of Chance, The Nature and Origin of Biological Evolution. Dr. Kunin is a member of the National Academy of Sciences and also serves on the editorial boards for major scientific journals. Ladies and gentlemen, Dr. Kunin, over to you, sir.
Hello. Oh. Hello? Hello, good afternoon. Many thanks to the organizers for inviting me to speak at this meeting, or at this wonderful meeting. I really regret I cannot be there in person. I was very much looking forward to visit India for the first time in my life. Unfortunately, this did not happen, but I must not apologize. I believe I think it is for the new, I think it's for the new American administration to apologize for this. But in any event, let me start sharing my screen. It would be
recording in progress.
It would be very helpful if someone could confirm that you can see my slides and hear me.
Eugene, can you confirm that you can hear us when we're using this mic? Eugene, can you confirm that you can hear us?
Yes, now I can hear you very well. I think it was not working properly for a few minutes, but I think now we're in business.
Okay, that's great. This is Karthik here, Eugene. Thank you so much for talking, and you can
yes,
take it away now.
I can hear you very well.
Excellent. So you can take it away, and we will get back at the end of the talk with questions.
All right, so today I'm going to speak to you about the world of viruses and its evolution as we now see it through the lens of metagenomic and metatranscriptomics. I was immodest enough to put here this epigraph from Albert Einstein to emphasize the unfortunately, I hear my own voice with some delay, which makes it very difficult to speak. All right, let us nevertheless try to proceed. So, let us nevertheless try to proceed. So I begin with this old data on the abundance of viruses in the biosphere to emphasize that viruses are widely to emphasize that viruses are widely considered to be the widely considered to be the most abundant biological entities on Earth. And it has become more or less a rather common meme that there are up to 10 virus particles per cell in many habitats. And the total number of virus particles that are present at any given moment reaches really hyper astronomical numbers like 10 to the power of 31. More done by my colleague and friend Puri Lopez Garcia from Université Paris-Saclay, using metagenomic analysis of viral hallmark genes, which we show. Which we shall discuss in a few moments, shows more modest data, and I think more modest ratios of viruses to cell, and I think more accurate, but nevertheless confirms the understanding that viruses are the most abundant biological entities on Earth. And by the way,
Eugene, sorry to interrupt you. Are you still hearing the echo?
In a beautiful previous talk by Dr. Petrich, they have heard about the enormous ecological role of viruses, in particular in soil ecology, so that clearly, yes. Right now, no. But until the last moment I was. Yes, I'm still hearing the echo.
Eugene, can you confirm that you have closed the YouTube link?
Yes, I'm afraid I'm still hearing the echo. I still hear the echo, and it makes it very difficult to speak. Yeah.
Eugene, could you maybe try taking your headphones off and only talking? That might help.
In a moment, let me end show for a moment.
Eugene, if you tried setting your speakers off.
Yes, I am sorry. I think I had it open elsewhere. I think now everything is okay. I'm very sorry about that. Yes, so we can now proceed. My apologies for this. It was actually my fault. I would like to remind you of textbook knowledge. I must not spend much time on this, but reminding you that unlike cellular life forms that all have large double-stranded DNA genomes and use the same scheme of information storage and transmission, viruses use all possible forms of nucleic acids as their genomes packaged into virus particles, suggesting the possibility, which we will not discuss in detail today, it would be another lecture, that viruses are actually nature's original genetic test systems. And I'm sorry about all these technical issues. Give me 1 second. I will just get my laser pointer. So an important message that I want to convey right now is that we now are fully convinced that there is no common ancestry for all viruses, not a single universal gene. So we cannot be phylogenetic tree or comprehensive phylogenetic tree of viruses as we can for cellular life forms. However, unfortunately, there is a certain rather small number of viral hallmark genes, genes that are, in quote, proteins performing the key roles in viral replication and particle formation, which are shared by large subsets of viruses, in particular those that have different forms of genome. And these allow us in the entirety, when we study evolutionary histories of each of these hallmark genes, we can get a picture of the evolution of the entire world of viruses. And now I must introduce the current megataxonomy of the world of viruses that has been adopted recently by the International Committee for Taxonomy of Viruses, and which consists of several of these realms, which is the highest taxonomic rank that has no direct equivalent in the taxonomy of cellular life forms, but more or less equivalent to a domain of life. And 4 of these realms include the great majority of known viruses, and in all likelihood the great majority of all viruses. The first of these is the realm Rizosphere, which consists of 2 kingdoms of Artornavir, viruses that whose whole life cycle is based on RNA, that don't use reverse transcription as a stage in their life cycle. The second kingdom, Pararnavir, that do use reverse, it's called reverse transcription, do use reverse transcription. Monodnaviria, small RNA viruses with circular genomes, typically with 4 kingdoms, and 2 vast realms of viruses with double-stranded DNA genomes. We shall talk in some detail soon about 3 of these large realms. I must emphasize that each virus realm is considered monophyletic based on the conservation and hallmark genes. And there are also deeper connections even between the realms based on sharing particular highly conserved domains. Now I will go very quickly through a few slides, just fleshing, if you will, the taxonomy within each realm to make the point that it has been worked out through the taxonomic rank from the realm down to virus families. So this Rizosphere, Monodnaviria, Duplodnaviria, of which we'll talk in greater detail, and Varidnaviria, a great diversity of viruses with double-stranded DNA genomes, which we will also discuss. So, as I said in the very beginning, we are looking at the world of viruses through the lens of metagenomics and metatranscriptomics. And here just a few remarks to get us on the same page on this. So the amount of metagenomic sequences now vastly, by orders of magnitude, exceeds the size of the regular sequence databases such as GenBank. I should note that on many occasions, the viruses are actually ignored or missed in metagenomic survey studies, unless they are the goals. There are 2 types of viral metagenomics, broadly speaking. When we are looking, when the researchers are looking at virus-enriched fraction, or more broadly, when we are fishing for viral genes in general metagenomic data. We will discuss projects based on each of these approaches. Generally, metagenomes have been, with considerable success, used to explore the diversity and biogeography of viruses and finding new genes and genomes. We will talk about this in a moment. A few additional remarks on viral metagenomes. So, there is good and bad in reality. Viral genomes are, generally speaking, small. Small, of course, means also the variance of 3 orders of magnitude, but nevertheless, the majority of viral genomes are small, up to a few hundred kilobase. And so with the modern methods of sequencing and assembly, it's very common to see a complete viral genome in assembled metagenomic data. Usually, we identify viruses in metagenomes and metatranscriptomes by detecting hallmark genes. But the difficulty, the flip side of that coin, which is now obsolete, we don't have pennies anymore in the United States, the flip side of that coin is that viruses evolve fast, typically much faster than cellular life forms. So finding homologs of even hallmark virus genes is a difficult exercise that requires the use of most sensitive sequence and by now structure search methods. And I will not return to this much, but really these sensitive approaches underlie everything that I'm going to present in the rest of this talk. But to say more about metagenomics and metatranscriptomics, which I like with respect to viruses called jointly metaviromics, is that this is now the principal approach for new virus discovery. Essentially, by far, many more novel viruses are being discovered by metagenomics in a relatively, and metatranscriptomics in a relatively unbiased manner that are identified by traditional laboratory approach. So much so that a few years ago, in this consensus statement, the International Committee for Taxonomy of Viruses, traditionally very conservative organization, has agreed that now virus species and higher taxa can be recognized solely based on the analysis of metagenomic data. And when we look at this slide, also somewhat out of date, we will easily realize why. Because what it shows is the identification of different viruses in various places in the world ocean by the Tara metagenomic expedition. And what is here on the horizontal axis is the prevalence of different viruses, that is in how many points of sampling points in the ocean they have been discovered. And here is the abundance, the number of reads, simply put, on logarithmic scale. And what is shown by these colored circles are viruses with known hosts, primarily bacteria. And the gray little circles are those that are found only by metagenomics. And you can easily see that both the most prevalent ones and by far the most abundant ones are only known from metagenomic data. And moreover, as I said, this is outdated. This is a few years ago. If I or someone else bothered to update this, you would practically not see the colored circles. They will be drawn by the gray ones. So the approach to discover novel viruses now and to chart the virus world is indeed metaviromics. Having introduced all this, I will now start with the new view of the RNA virus field. That is, recall, the viral real riboviria. So in this study published a couple years ago, they analyzed some more than 5,000 metatranscriptomes with more than 1 billion context and detected segments encoding the hallmark gene of RNA viruses, the RNA-dependent RNA polymerase, PRP for short, by using, as I pointed out, sensitive methods for profile sequence search. The result of this was rather dramatic in that more than 500 new taxa at the higher than family level have been discovered all over the globe, and a variety of novel domains have been identified in uncultivated RNA viruses. Qualitatively, the most important message to which I will return in a moment is the discovery of new RNA bacteriophages. Prior to this work, the known knowledge of RNA bacteriophages was the knowledge of RNA bacteriophages was very limited. Only two narrow groups were known. Now we show that there are many, many more of RNA bacteriophages that have been simply overlooked, and some of them are even targeted by bacterial CRISPR systems, so clinching the case of the bacterial host. So I just want to emphasize once again, without going into every detail, that for discovery of RNA viruses, what we do is extensive search for RNA-dependent RNA polymerase in metatranscriptomes. And the result of this particular study, there are several parallel complementary studies, I must say, was about a five-fold increase of RNA virus diversity at every taxonomic level, except for 5, only, which is the highest taxonomic level in this context, of which only a couple have been tentatively discovered. But at every other level, and this is very high level of taxonomy, like classes, a five-fold or more increase in the diversity has been identified. Coming back to RNA bacteriophages, here in our previous analysis, phages comprise only about 3.5% of detectable RNA viruses. Now, the increase in the contribution of identifiable RNA phages is tremendous. It's more than 1/5 of RNA viruses. Now, with a couple of putative new phage file, and we shall see that more can be discovered, and probably more RNA phages hiding here among the viruses that previously were considered to be eukaryotic. So, these are the broad surveys of the RNA virus field. New frontiers, distinct groups of RNA viruses found by metatranscriptome analysis of particular habitats. Our very recently published paper with the Japanese colleagues, where an ingenious sequencing method was applied to obtain complete sequences of multi-segmented RNA virus genomes. And a couple of novel groups can be discovered in a habitat that is not generally considered as hospitable to RNA viruses, that is hot springs, but nevertheless, we now realize they are very much there. And one of these was particularly interesting in that they were found to encode unusual RNA-dependent RNA polymerases, which whose structures are modeled here and compared to the known structures of RNA-dependent RNA polymerases and reverse transcriptase that appear to be intermediate between typical ribovirus RDRPs and reverse transcriptases. So this is now a new phylum in Riboviridae, which may be upgraded to a new kingdom, showing that major novelty remains to be discovered. And now I have to quote this very recent paper by, not ours, by Eddie Holmes and coworkers, who, in the spirit of the time, applied artificial intelligence methodology to analyze the hidden RNA virusphere, an even greater number of they analyzed an even greater number of metatranscriptomes, and discovered a very broad diversity of riboviruses, which we as a community still need to digest and classify. And this is a major challenge, but as I explained in the beginning, we already have the megataxonomy framework that hopefully facilitates that task. So as I said, we will go through different realms of viruses and see what metagenomics can do and metatranscriptomics can do and does. And here is something that I think is very strange. And that is the discovery of the most abundant human-associated virus of all. The most abundant human-associated virus is not, by any account, influenza or SARS-CoV-2, but it's a particular variety of bacteria, which was completely unknown and uncharacterized. I shouldn't even say uncharacterized, completely unknown until the advent of advanced metagenomic approaches. And then it has become clear that this virus called cross-stage form, cross-assembly, is dominant in human gut virals, found in 50%, if not more, of the gut viral samples, where it can comprise staggeringly up to 90% of the viral sequence 3. This was discovered by Rob Edwards already a decade ago, and at that time this virus looked unique. And moreover, what was worse in a sense was that the proteins encoded in this genome showed no significant similarity to available protein sequences, the great majority, precluding the classification and understanding of that virus. So my colleagues and I tried to remedy that situation, again, by search of metagenomic database. And that was successful in the sense that we identified several hundred phage genomes that were at least different levels, to a different degree, related to cross-phage, the great majority of them coming from metagenomic and fortunately from metagenomic data. And fortunately, we managed to identify the hallmark genes of the real duploid Naviria, describing what looked at the family of grass-like viruses at the time. Here is the narrow cross-phage group, the original cross-phage, and here is a broad variety of diverse viruses that, however, share common genes with this cross-phage and can be confidently classified into this family, as illustrated here by the phylogenetic tree for major capsid protein. We continued that work, discovered now thousands of highly diverse viruses that now comprise the new phage order Cross-Viralis, the first phage order formally recognized in the new taxonomy. The great variety, if not all of these viruses, associated with bacteria of the phylum Bacteroidetes, which is the major inhabitant of our guts. And without going into detail here, some of the members of this order have very unusual biological features with alternative genetic codes used, with genes interrupted by all kinds of model elements, self-classic introns and inteins, with frequent exchange of DNA polymerase genes between closely related branches, I would like to emphasize that this metagenome mining is not just busy work. It helps us discovering major novel biological features and also venturing into the structural analysis of these remarkable viruses, on which we collaborated with our colleagues at the University of York in England, producing sort of a structural atlas of a class phage, which shows remarkable, unusual features with many cargo proteins along with the genome incorporated into the virus particle and injected into the cell with the phage genome as controlled by the specialized, unique, so-called muscle protein, according to this scheme. So again, this is the point that I want to make, that these metagenomic analysis combining sequencing and bioinformatics lead to biological insights. I will now switch to the other vast realm of viruses with double-stranded DNA genomes, Varidnaviria, and in particular to the largest phylum in this viral realm, Nucleocytoviricota, which includes the giants of the virus world that shown here. Viruses with genomes of up to 2.5 megabase, more than the genomes of maybe half or more of bacteria and archaea, known as Pandora viruses, and probably viruses with even larger genomes remaining to be discovered, leading to ideas of origin of these viruses from cellular life forms, but probably not very realistic ideas. We realize clearly by evolutionary analysis that these very different viruses have a common origin and giant forms emerging independently in many branches of the phylogenetic tree. But what I want to discuss here once again is the discoveries made by metagenomic analysis, to begin with the small relatives of giant viruses in the phylum Nucleocytoviricota, which we call MRIA viruses. In a timely association, the word Maria means a dream in Ukrainian. So, with a quick introduction to the smaller relatives of these giant viruses, this is a family of crustacean viruses that, for a change, has been identified a few years ago by traditional laboratory techniques, and which are the smallest of Nucleocytoviricota, with genomes of 70 to 75 kb. And this is again a virus, a very mysterious virus that has been isolated by and colleagues in Brazil. A small viral genome, about 45 kb, encoding 74 proteins that could not be properly classified at the time as a relative of any known viruses. A little later, it has become clear that partially sequenced genomes of this infecting this associated with this alga, unicellular alga, are relatives of that virus, but this is where it stopped until we started dedicated searches for capsid proteins and packaging ATPases of these viruses and metagenomes. And lo and behold, we are lucky enough to discover a new group of viruses related to nucleoside replicata. I will show in a moment. Well, actually, you can see here how they're related in these phylogenetic trees of two hallmark proteins, and forming these two distinct subgroups. There are already many genomes of these tiny relatives of the giant viruses. They have their own set of hallmark genes. We identified hallmark genes at different phylogenetic depths or different levels of taxonomy. So they have a set of 10 shared conserved proteins. And most interestingly and uncharacteristically, in general, of viruses with large double-stranded DNA genomes, they encode proteins related to homologous to rolling circle replication initiation and the nucleases of single-stranded DNA viruses. So they probably have an unusual mode of replication that remains to be investigated, continuing the theme of biological discoveries for metagenomics. And then we look at these core genes. We get a clear connection to nucleoside vericota, looking, for example, at this transcription factor, which is not found anywhere else in the world. So these viruses probably form a novel class in the phylum nucleoside vericota. We will come to the taxonomy in a moment. Now I want to introduce something in addition. And this is a discovery of so far mysterious giant viruses in nowhere else than in our own gut microbiomes that have been surveyed for the presence of members of nucleoside vericota in our joint work with the colleagues in France, in Genoscope and in Rizosphere. And what has been discovered here is a novel group within this class of the awkward name Pockets viricetes, which so far included two viral orders. One is pox virus, including the agent of the devastating human disease, now eradicated, fortunately, smallpox. And the other is asper viruses, including a virus that is very, very important pathogen of pigs, African swine fever virus. So in this analysis, a novel order apparently was discovered, which includes these egoviruses with their own large set of conserved proteins, very large genomes that are common in the gut metagenomes of a variety of vertebrate animals, including very much ourselves. So again, until dedicated pipeline for discovery of nucleoside vericota members has been developed, these viruses remain completely under our radar. And they're interesting, apart from being interesting as inhabitants of our guts, they have a remarkable structure of capsid proteins with a huge giant insertion here compared to other members of the nucleoside vericota. So the taxonomy changes, as we discussed in this recent review article, with the Maria viruses becoming probably a new class that may be as compatible with their small size, may be ancestral to all nucleoside vericota, and egoviruses becoming a new order within one of the classes of this phylum. Let's come back now to the megataxonomy and mention two new realms of two new, much smaller, or at least so it seems, realms of viruses that have been delineated soon after the four major. One of these is adenoviria, a very interesting compendium of viruses infecting archaea, initially found only in Archaeohyperthermophiles. Now we realize infecting all kinds of archaea which incorporate a form of DNA into the virions, which was not suspected until these viruses were studied in detail. But these I'm not going to discuss. What I'm going to talk about is other little realm called Rizosphere, which at the time it was established some 3.5 years ago, consists of a single representative, an important one, hepatitis delta, hepatitis D virus, which is a depends in its reproduction on hepatitis B virus and causes especially severe forms of hepatitis in humans. And the genome of this virus is very interesting. It is a small circular RNA of about 1.7 kb, which encodes a single protein, viral nucleocapsid protein, nothing else. So in everything else, this virus relies first on the post-enzymatic machinery, specifically transcription machinery, and second on catalytic RNA modules encoded in its own genome. So, we will now talk about the realm of viroid-like circular RNA. Maybe even more than one realm. Viruses. But first I must introduce viroids and related RNAs. Viroids may not be particularly widely known, but I think they really deserve to be. Because they're the smallest known pathogens and broader, the smallest known replicants, which are tiny circular RNAs of only 200 to 400 nucleotides, which have this highly stable, typically have this highly stable secondary structure. And despite, which encode no proteins, but despite their tiny size, can cause diseases in plants, for which they have been identified. And there is a sort of a family of viroid-like RNA agents, some of which are satellite RNAs, satellites of plant viruses. The others are the ribosi viruses, hepatitis delta and its relatives that I have already mentioned, and yet others are so-called retrozymes, which are viroid-like RNAs encoded by small transposons. So the tiny size of these replicants, viroids and viroid-like RNAs, somehow implies that these might be relevant for understanding the primordial RNA world, for understanding the origin of life. However, this idea proposed a long time ago was not taken very seriously because of the narrow host range of these agents. They have been, for many years, they were considered to be confined to a few plant species and therefore not good candidates for a role in the origin of life. So you, my colleagues, we were interested to try and find out whether this really reflects the spread of such agents in nature, or alternatively, they have been overlooked. And so we started mining the metatranscriptomes. The same collection of metatranscriptomes that I mentioned previously that we use for discovery of viruses, of regular viruses. But now we developed a dedicated pipeline to identify covalently closed circular RNAs. And indeed, most remarkably, these small covalently, and not so small as we shall see, covalently closed circular RNAs have been discovered in samples from a broad variety of diverse ecosystems, including many that essentially contain no plant or animal material, suggesting that the actual host ranges of these types of agents are much broader. Altogether, like in the study of RNA viruses, we obtained more than 5-fold expansion of the known diversity of viroid-like RNAs. And interestingly, we again found that some of these viroid-like RNAs are targeted by CRISPR space, by CRISPR systems containing reverse transcriptase, hence capable of targeting RNA genomes, indicating that some of these replicated bacterial hosts. I will say a little more about this in a moment, but I would also want to emphasize that in the process of this study, additionally, a broad variety of viroid-like viruses coding various proteins have been discovered. Some related to ribosome viruses, hepatitis delta virus, some unrelated. So there was about a 5-fold expansion of the diversity of viroid-like covalently closed circular RNAs. I must indicate that in this analysis we have been very conservative, considering to be viroid-like only those covalently closed circular RNAs that contained readily identifiable ribozymes. Ribozymes, so that we are on the same page, are catalytic RNA modules, little domains of RNA that possess self-cleaving activity and therefore are involved in the processing of replicative intermediate of viroids, hepatitis delta, and related agents. With this conservative, a lot had been discovered by this conservative, but that left uninvestigated enormous amount of dark matter that may or may not contain novel types of ribosomes, which remain to be investigated, and we are working on that. Now, for what has been discovered, though, with the conservative, there already has been a huge increase in the diversity of ribosome viruses. Viruses that with circular or covalently closed RNA genomes that encode distant relatives of the nucleocapsid protein of hepatitis delta virus with this peculiar structure. So in this tree based on the alignment of the sequences of these proteins, here is hepatitis delta. Here is distant relatives that have been discovered in several studies in various other animals, vertebrates, and insects. And here is what we have discovered in meta-transcriptomes. The real diversity obviously is here, and in all likelihood, not just the diversity of viral-like agents, but the diversity of hosts as well. Some of these entities are very interesting and sort of challenging for classification. These are the so-called MBI viruses, which are associated with fungi, probably infecting fungal hosts, and that are viroid-like in their organization, but quite large for this part of the virus world, about 5 kb, and carry 2 protein-coding genes, one of which encodes an RNA-dependent RNA polymerase, which is homologous to the RNA-dependent RNA polymerase of members of the real Riboviria, although not closely related to any particular group, presenting challenge to understanding of the evolutionary classification, because these are sort of chimeric between viral-like agents and regular RNA viruses. Right now, based on the presence of the RNA-dependent RNA polymerase, they have been classified as the new phylum within the kingdom Artornavir. Very recently, in this remarkable work published by the laboratory of Andy Farr from Stanford, these bacteria, viroid-like circular or covalently closed RNAs have been identified, believe it or not, right in our guts. They have been called obelisks. They're circular, they have circular RNA genomes which encode a single, completely uncharacterized protein without homologs detectable anywhere else. But importantly, some of these agents, probably can call them viral-like viruses, or RNA plasmids perhaps, have been associated with distinct bacterial isolates of Streptococcus in particular, showing that there are wonderful surprises in the hiding in the viromes in our very own guts. So from all these analyses suggest that the evolution of viroid-like agents is sort of a two-way street. We have multiple interconversions between non-coding tiny viroid-like things and riboviruses encoding RNA-dependent RNA polymerase and other genes. And this, I think, suggests resurrecting the idea that viruses can actually be relics of the primordial RNA world. Before closing today's lecture, I want to leave with this little, more or less back of the envelope estimate of the size of the global viral genome, using the conservative assumption on the number of bacterial species in the biosphere. There are probably more, and conservative assumptions on the number of viral species associated with particular bacterial hosts. Some are known to host many or more, and some realistic estimate, range of estimates on the number of unique genes in a large viral genome, such as typical bacteria phages. This leads to this astronomical estimated number of unique viral genes on Earth, which is likely to be at least an order of magnitude more than the number of genes of cellular life forms, at least current, suggesting or indicating, I would say, that viruses comprise the principal pool of genetic diversity and genetic novelty in the biosphere. And this is where I stop just with these take-home messages. Viruses are ubiquitous, the most abundant biological entities on Earth, and the main pool of genetic diversity, and the fundamentally important ecological agents. Metaviromics now is a true revolution in the study of virus diversity, and by far prevailing approach in the discovery of new viruses. Nowadays, we are settling, or maybe settled, on the global megataxonomy of viruses, but major new groups of viruses, including new kingdoms, new realms, and major inhabitants of our guts, in particular, are being discovered and remain to be. So, what the principal message that I wanted to convey today is that the new virus discovery, metagenomes, metatranscriptomes, informs, in most interesting way, our understanding of the structure and evolution of the virus world. And I finish with acknowledgement of members of my own lab, including the very talented student Benjamin Lee, who worked on the viroid-like RNAs, and my key collaborators, in particular, Valery Tolev from Oregon State University and Mark Turpovich from Institute Pasteur, and collaborators from all over the world. I could not, unfortunately, give credit to everyone who has been involved, but the main collaborators are mentioned here. Thank you very much for your attention, and I will, although I realize that I'm probably the only thing between you and the poster session, drinks, concert, and some beautiful Indian dinner, but nevertheless, I'm happy to take questions.
Yeah. Questions from the audience? Okay. Thank you, Dr. Kunin, for that fantastic talk. I have a quick question. Are there any practices to enrich for viroids from environmental samples? Am I audible?
I'm unaware of anything actually done to that effect. I believe new methods need to be developed to enrich for this. It's doable, of course, but I don't think that these methods have been developed or at least widely used. I think this remains to be done.
Great. Thank you. Thank you so much for that fascinating talk again. Thank you for joining us. And thank you, ladies and gentlemen. That brings us to the end of this session. Thank you very much.
Thank you.